Good morning. Sir, with your permission, should we begin? A small request, if we all can switch on our cameras, but stay muted. Thank you. Do you know that biotechnology traces its roots to the Neolithic era, when early civilizations discovered how to ferment grains to prepare alcoholic brews and bread? It wasn't until genetic engineering came mainstream in the mid-1970s, however, that biotechnology as we know it today was launched. The field continues to evolve today. The advent of new technology has revolutionized the market in recent years during industry expansion. One of the chief factors driving market demand for biotechnology products is the growing incidence of chronic ailments such as diabetes and cancer worldwide. Recently, of course, COVID-19 has emerged as a therapeutic focus. The massive efforts to develop vaccines, deploy test kits, develop highly predictive antibody tests, repurpose existing antiviral drugs, and develop new drugs are causing billions of dollars to flow towards many biotech companies. Even the new national education policy emphasizes that India must take lead in preparing professionals in cutting-edge areas that are fast gaining prominence, such as biotechnology, genomic studies, nanotechnology, neuroscience, etc. Good morning, ladies and gentlemen. I'm Dr. Bhavna Gupta, Assistant Professor at School of Biotechnology, KIIT University, Bhubaneswar. I welcome you all to the Biotech Industry Conclave, emphasizing on alliances and deputations, empowering biotechnology. To set the stage right, I now invite Dr. Srinivas Patnaik, Dean, School of Biotechnology, KIIT University, to say a few words and help us understand how the academia-industry alliances are important for us to build a better tomorrow. Dr. Patnaik, please.
Thank you very much, Dr. Bhavna Gupta, for giving me a chance to speak a bit. Good morning, one and all. It's a great pleasure to welcome you all to this wonderful platform of Biotechnology Industry Conclave 2021. I would like to take this opportunity to thank and welcome today's speakers, Dr. P.M. Murali, President, Able Business Council, and Professor Anil Kaul, Vice President, Johnson & Johnson, and Professor in London School of Hygiene and Tropical Medicine, and all the panelists from industries and academia agreed to participate in this conclave. I'd like to welcome our own respected Pro-Vice Chancellor, Madam Professor Susmita Samanta, and respected Professor Mrityunjay Suar, Director General, Research and Development, KIIT University, and CEO, KIIT TBI, to address the gathering. My dear faculty colleagues, staffs, and most importantly, dear students. Parallel to the visionary notion of Professor Achyuta Samanta Sir, founder KIIT and KISS, eradication of poverty with education, this kind of congregations closes the gap much significantly. To say a few words about the School of Biotechnology started in 2007 by Nobel laureates Professor Richard Ernest and noted immunologist Professor Hans Hengartner, Nobel laureate Rolf Zinkernagel. Under the vision of Professor Achyuta Samanta, founder KIIT and KISS, and the leadership of Dr. Mrityunjay Suar, it has come a long way with several accolades and milestones. With humble beginning with only one MSc in biotechnology course, we now are running 3 different streams, that is Masters in Biotechnology, Applied Microbiology, and BTech, MTech dual degree in biotechnology. And slowly we have spread into the PhD and postdoctoral programs in biotechnology. The school has awarded with many institutional grants and accolades. That is, to name a few, it is DBT Builder Program, FIST Program, and Center of Excellence Award by Department of Biotechnology and Department of Science and Technology by Government of India, with 52 running research grants And that is leading to publishing in international peer-reviewed journals and high-impact factor. School of Biotechnology is doing tremendous job in bringing science in academia. The school focuses on translational research leading to technology development, innovation, and patents to its credit. We at School of Biotechnology have realized the significance of industry-academia alliance, hence come up with this Biotech Industry Conclave since 2014, wherein big wigs of industries are invited and have discussions and deliberations. Their exercise benefited the student, academic, and scientific fraternity as it inculcates the flavor of industry culture and the reality behind industry needs. The Biotech Industry Conclave platform provides immense opportunity for our students to have industry connect through discussions and interesting question and answer sessions. We also experience technical troubleshooting requirement of the industries, which will be addressed by the scientists. So this is a wonderful opportunity for this crosstalk between industry and the academia. The theme of this year's Conclave Alliance and Deputation, empowering the backbone of biotechnology. The conclave will have keynote address followed by 4 sessions wherein various industry delegates will present their views, thoughts, and their suggestions. Thank you very much for gathering in this particular platform. I welcome once again everyone for this Industry Biotech Conclave and congratulate all to be part of this particular congregation. Thank you so much. Over to Bhavna, madam.
Thank you so much, sir. The dreams of biotech industry to evolve financially and professionally cannot come true without building alliances and mergers. That's well said by you. And recently, we know 5 of our domestic pharma majors have joined hands for a clinical trial of an antiviral drug. Let's now hear from our Dr. Mrityunjay Swar, Director General R&D, KIIT University, and CEO of KIIT TBI Incubator. Sir.
Thank you. So thanks everybody again. Let me welcome our guru here, Dr. P.M. Murli, who is from the ABLE, from the industry, and ABLE is running the whole biotech ecosystem in the country, advocating for the better biotech sector in the country. I welcome you, Dr. Murli. You've been a prolific supporter, well-wisher of KIIT School of Biotechnology. With that, also you have Dr. Anil Kohli is going to join us soon, probably. And he has been one of the board member of us and always talks about translational things. So with nothing can be better than joined by these 2 eminent personalities today, Biotech Industry Conclave. Now, as Dr. Patnaik said, the School of Biotech has made its humble beginning, become a center of excellence. But this is also promoted a lot of startup companies. KIIT TBI was born out of that, and KIIT TBI has promoted several companies, and today are very popular in the country. Now, because the faculties at School of Biotech understood science, but they want to build technology out of it, what do you call biotechnology? We don't want to really build biosciences platform here. And let me be very honest with all of you, dear the speakers today, especially Dr. Murli, we are pro-industry oriented people. Like we take pride in inviting Nobel laureate, the same equal pride we take inviting the corporate leaders, because we understand in academia, if the industry, they don't come and they don't build authentic partnership or better engagement model, the academia is not going to do better, because we cannot build industrial platform here to train our students. So that's why I'm very happy that what we have started Biotech Industry Conclave is going on so smoothly every year. That has resulted us, building our curriculum better. And when we approached ABLE, especially to Dr. Murli and Mr. Krishnan of ABLE, the president, and they're probably very happy with our deliverables and achievements, and we requested ABLE to really help us to build up ABLE Center here. I think Dr. Murli is going to speak on that, how ABLE Center is going to set up at KIIT to help our students, faculties, and reach out to East and Northeast regions so that we can really build better human resources for the country. Now it has gone big now. So School of Biotech is one end, the other end's KIIT Technology Business Incubator. Then we have now building up Bhubaneswar City Knowledge Innovation Cluster. And all of you will be very happy to note that even the Biotech Finishing School, we are going to start soon. So that's also DBT has been kind to approve. One of the Biotech Finishing School will be needing the help of able to really build it better. But beyond that, also there will be biotech cluster is going to come up to Bhubaneswar, which is called Urjit cluster. So these are all going to happen in Bhubaneswar, in KIIT School of Biotech, in KIIT University, in KIIT BI, and I'll be requesting all the industry leaders, those who have joined here today, please come with an open mind. We are going to welcome you in a bigger way. We really want to work with you in a bilateral way. We don't want to be a parasite, that we only want to place students. The main purpose of industry conclave is to really talk to each other, cross-talk should happen, ideas should come out, and then we can really work together for a better, productivity or better outcome. I think I'm very happy that all of you have joined today, especially special thanks to Dr. Murali, because he has been kind enough to talk about us in various platforms. I think that's what I think I have realized. In many places, he talks about that there's a School of Biotech at KIIT, which is doing great, and he has been a better advocating for better biotech culture in the whole country, and he talks about KIIT TBI in many platforms. So with this, I again welcome all of you. I thank our Pro Vice Chancellor, Professor Smita Samant, who has been kind to really support both School of Biotech in a great way and KIIT TBI in a bigger way. So I think a lot of going to happen in near future, and KIIT School of Biotech has been a center of excellence, and soon it is going to be center of relevance for translational science, and we can really build several startups out of our spin-offs, out of our ecosystem. Thank you. All the best for the industry conclave. Thank you.
Thank you so much, sir, for your words. I think COVID-19 pandemic has the biotech sector's first modern challenge, similar to the development of maybe an atomic bomb or the race to the moon. And I also think that the industry's response to the challenge is likely to generate significant interest among new groups of students, investors, and brilliant minds. So let us invite Professor Sashmita Samanta ma'am, Pro Vice Chancellor of KIIT University, and hear her views on this fast-growing field of biotechnology. So, Sashmita ma'am, please. I think she got disconnected somehow, maybe. I request Gopal sir to see if she's available online.
I'm contacting her, and let's wait maybe 1 minute.
Sure.
Yeah.
So is she available? Has she been able to join back?
She joined a while ago. She was there, just trying to.
I think somehow she got disconnected. I think. Once she will join, maybe we can ask her to talk. But I think at this moment, let's start.
Okay.
Let's move on.
So if I may request Dr. Kapil Chaudhary to present the vote of thanks to the speakers and declare the official beginning of the Biotech Industry Conclave. Dr. Chaudhary, please. Dr. Chaudhary, if you may please present your vote of thanks.
Thank you. Good morning, ladies and gentlemen. So as explained by previous speakers, Industry Academia Alliance is the key factor in creating novel techniques and generating employable human resources. And in order to fulfill this, School of Biotech at KIIT organizes annual Biotech Industry Conclave. And here we are presenting 7th in the series, Alliance and Deputation, Empowering the Backbone of Biotechnology. A lot of people have contributed to make this event possible. And first of all, I would like to thank all the speakers who spared their valuable time to be a part of this conclave. Thank you, sirs and madams. I thank Professor Achyuta Samanta, founder of KIIT and KIIT, for providing this beautiful facility and infrastructure. You are always a source of inspiration for us. Thank you, sir. Big thanks to Professor Achyuta Samanta, Pro Vice Chancellor of KIIT University. For this, of course, she was supposed to be here. Of course, we expect she will be here soon. Thank you, ma'am. And your presence is always enlightening. Thanks to Professor Mrityunjay Swar, CEO of KIIT TBI and Director General R&D KIIT University, who also happened to be the founder director of School of Biotechnology. And thank you, sir, for always guiding us to the right path. Thanks a lot. And a big thanks goes to our student fraternity and faculty, our Dean, Dr. Patnaik, support group, ICT personnel, corporate relation team from the KIIT, and the student volunteer team of the Bio Nights for making this event a grand success. I'm sure all the students will draw a lot of benefit from this event. Thank you. Thanks a lot. Let's have a beautiful and nice conclave. Thank you. Over to you, Baba.
Thank you so much, Dr. Chaudhary. So on the behalf of School of Biotechnology, we declare the official beginning of Biotech Industry Conclave. And let's move on to the first session of the day. And I request Dr. Chaudhary again to take over so that we can begin with the first keynotes.
I think Dr. Patnaik will take over the session. Sorry.
Okay. So I request Dr. Patnaik, please.
So once again, welcome everyone to this Biotech Industry Conclave 2021. So today's our first speaker is Dr. P.M. Murali, President, Able Business Council. Welcome, sir, for this Biotech Industry Conclave. To say a few words about Dr. P.M. Murali, he is a serial entrepreneur for more than 2 decades with many first of its kind products and innovation in many areas of biotechnology, built pioneering IT greenfield projects, was part of a venture capital organization to evaluate prospective technologies for commercialization. He has nurtured and built the entire Indian arm of $100 million Swiss biotech company, India's first pioneering public-private partnership to set up country's first genetic chemistry laboratory inside a premier CSR campus, that is the IICT, built the R&D subsidiary for the $250 million Indian FMCG group pioneer in the world to conduct full-fledged clinical trials, all 4 phases, with polyherbal formulation for 7 disease indications over 2 decades. He is a great personality in the industry, biotech industry. So I would like to welcome Dr. P.M. Murali, President, Able Business Council Chairman, to address the gathering. Thank you, sir.
Hope you are able to hear me this morning. Good morning to all of you.
Yes, sir. Yes, sir.
Okay, so thank you for the kind words of introduction. I think I'm always very nervous when these introductions are done. You could have seen me struggling. So sorry about it. There are much more accomplished people in the world, and it's always something that I keep in my mind all the time. So I have a stop clock here that I have turned it on. So I will try to limit myself to about 15-20 minutes. I can see that there are close to about 215 participants on the list here. We'll be happy to take some questions here. So let me first thank all the Zoom luminaries and accomplished people who are on the Zoom dais today. So thank you very much. So let me begin with a very simple introduction about what I am going to do today and what is the stage that I am planning to set in today. So as you know that, some of us were part of the team responsible for building the blueprint as well as the vision for the entire Indian biotech industry. It was pretty nascent. A lot of us did not have any clue on what number we should put there. We had no idea what was the annual growth rate that the country was actually going through. And how can we even look at a number? It was in this context that almost about nearly close to 12 to 13 years ago, that we decided to take the bold step of making a roadmap for the biotech industry and see what happens. So we started with a very simple concept that we were currently growing at about 15 to 18% as a industry, and said, why not we look at slightly improving that percentage of growth to reach about $100 billion in about 2025 time frame. I think we are somewhere in the 70 or $80 billion at this point of time, and we still have a few more years left on that. So we are on track to achieving something much more. And so what happened was, even when we set the $100 billion, people said it's a stretched goal. But then, as you know, when you have a goal, you don't know whether it is stretched or understretched. But the point is, you will have to make it somewhere, and you will have to reach it somewhere and somehow. So this was one of the initiatives that happened. And right now, the goal has moved to about $150 billion, largely due to the extraordinary efforts that the biotech industry put in during the pandemic. Now, there is a learning all the time, and all of you would have heard enough and more about this pandemic and what it has caused. But what you may not know is that during the pandemic, a lot of us worked from our homes on trying to ensure that the industry still continues to function and take up the challenge. And there was a task force that was formed. There were several small task forces that were formed and so on. And the beauty is, all of us had a role to play in trying to see how we can get this across. So things like approvals, things like personal protection equipment, things like kits for PCR, the PCR itself, primers. There were 100 other things that were missing in the country. But remember, all through the way, all through the pandemic, including the vaccines, including the material that is needed, including detection kits, including simplification process, including the way medical people were going to treat patients, all evolved during the pandemic. Now, this is something that we have to pat ourselves on the back as biotech industry, because it sounds very simple, but you have to remember it is not so simple. If a lot of us are alive today, a lot of us are able to communicate and get out to the other side of the pandemic, we should give a very, very big thanks to the biotech industry for taking us all there. Now, you have to remember, there is no country that can make progress if there is no leadership. And I would say that leading through tough times is the hallmark of a great leader. I am apolitical, so please don't get me wrong. I do not belong to any political party, but I have been completely taken in by the amount of efforts the current Indian Prime Minister had put in, the amount of importance he gave for Indian vaccines, the amount of money that was sanctioned through Dr. Renu Swarup's department for a number of projects. And now currently the Minister, Mr. Jitendra Singh, Honorable Minister Jitendra Singh, is also taking the bull by the horn in trying to push this industry forward. Now, on the same note, I would like to transition to your local leadership, which is under Mrityunjay. Now, I would say that when he first called me, I had no idea who he was. And he said, we are starting something in Orissa. So my first reaction, I am generally a very composed person on such things, but my first reaction was really an awe. I said, what? Orissa? So that was the last place in the universe where I thought biotechnology will be there, even though I have been working in close coordination with Central Rice Research Institute when I was a student. But I never thought that there will be something here. And I think all of you in this room, Zoom platform room, should feel very, very privileged, happy that one person along with his team, without a team, I'm sure Mrityunjay could have never pulled this off. But I would say that it has been a stupendous journey to where he has brought this institution along with all of you, because it is now very much in the map. I only simply go and talk to people saying that, Mrityunjay has done it. There is this extraordinary cluster in Orissa and so on. So I have done nothing more than that. But I think the full credit should go to him. So on the same note, I would like to say we escalated this during our meeting at the industry body. And everybody felt that if at all there is a reason why we should have a chapter of the ABLE sitting in this city, Bhubaneswar, we thought we should take the assistance of your institution, the assistance of Mrityunjay, and all the other like-minded people to see that there is a very, very good industry and an academia interaction there. So the framework has been completely drawn. As you know, every institution has fiduciary obligations that these ideas will have to pass through AGMs and other things. And AGMs, as you know, are now postponed due to COVID pandemic. So sometime around November, we expect all these resolutions to be carried through in our association AGM. And hopefully the year 2022 will see us present there. And we will also go to the next stage. Okay, so I will take just a few minutes on explaining two things to you, which I thought is very important as students on the other side. It was during my time, and it was Dr. Natesh, who was responsible from Department of Biotechnology. We both single-handedly pushed as much as possible, along with all the other past presidents and the current presidents, to see whether we can have a student competition. And the student competition was modeled on several ways, and finally we narrowed down on what we call as the BEST program. Now each center almost does a BEST program, but that was the only national level program at that time. And we had a fantastic jury. We had a fantastic set of SOPs put in there. You will be quite surprised that there are several companies that have now grown, matured, and come up to a stage on account of this one single program called BEST, including companies like C6 Energy, which is now doing pretty well. So the whole atmosphere of industry and the academia was actually fostered by this one program, which is called BEST, where we brought in investors, we brought in the industry people, and then we managed to get the students to present it in a very nice way. I mean, these were all the early days, right? I mean, after that, there is BIRAC, and there is BIG, and there is Sparsh, and there are hundreds of such things that now Mrityunjay is also running every day. But this was the seed, this was the platform for the students to actually come up and form teams, form associations, form alliances, which is the most important part. Now, I think this year's theme is absolutely wonderful, because without the coordination, cooperation, alliances, there is no way biotech industry is going to serve anybody in the world. Now, we have learned from the pandemic that the world is now just one. Nobody escapes from anything, irrespective of whatever border you may have, whatever army you may have, whatever navy you may have, you still have it porous to biology. So it obviously means that we need to be together to solve a problem here. So I think this year's conclave's theme is bordering on that. So we'll be very happy as an industry team to always visit your center to take it forward. The second part is more on technology part. I think this is what the students will be very much interested to know. So you heard all the magnificent things that I built, all of them into several $1 million, etc. It's very flattering, but my as you know, when you create wealth, it is for the investors. You don't create much of wealth for yourself unless there is somebody who says, yeah, you created all this, no, take some portion of it. But the key to this wealth creation is great ideas. You just cannot have run-of-the-mill ideas. You cannot have very, very inferior science. You cannot have something that is very, very, Me Too type of ideas. What you need is absolutely cutting-edge ideas. Now, some of us have lived like that. The adrenaline has to pump in every day in our head. We should feel very elated and so on and so forth. So you see my age. I'm so happy today because last night, I was so tired. I had so much of work to do, but then the meeting for me was fixed from California. At 9:50 was my first pitch session, and at 10:10 was my second pitch session. And what was I supposed to do? I was supposed to go along with my colleague on a Zoom platform to explain to Berkeley, where all the top scientists of this world are sitting, on a concept that we had sent to them. And I am extremely pleased to say, out of 700 companies, and mind you, 700 companies from the United States, where you have so much of ideas, my company is now in the finals of that idea. Now, the sad point is, if I present this idea to an investor in India, or if I present this idea to somebody in our Indian funding agency, it will be very interesting to see how they react. See, this is one of the things that Mrityunjay and the team will have to remember is that great ideas can come, great new technologies will come. Are your evaluators capable of looking at the opportunity? That is where industry will play a very, very big role, because we see what's there through a crystal ball. We may be right, we may be wrong, but we take those bets to go to the next stage. Now, that is something that every one of you should learn. So that's the subtle difference between how academia and the industry is, because industry will take those bets and it will move towards frontier science, and it will not hesitate in doing so because it will feel that that's how the future grows. And that's exactly is the segue to how mRNA vaccines came into being. If you remember, prior to the COVID, there was not one mRNA vaccine or one mRNA product was even approved by FDA. And you will be very sure that there was nothing like that. So everybody said, oh, no, this is not going to work, that's not going to work, etc. Then what happened? The pandemic completely changed the whole perception. Today, that particular branch of doing RNA vaccines is one among the topmost ideas. And also it is now moving into cancer treatment, it is moving into other segments, and so on and so forth. So the key is, how can you actually put in a lot of new ideas and thought process? So I'll spend the next 5 minutes on one of the ideas that we put in, which is what Berkeley felt that we were among the top 10 people out of the 700 companies that came. So I hope all of you know that you are just only 1% human, and the remaining 9% is microbes. So this may be quite startling to you, but the fact is that's true. So while you say this is Murali, I am 99%, 90% made up of microbes, and only 1% is Murali. So which means there are almost 10 times silent people living in me. And this 1% or 10% Murali is making all this big noise, whereas all the other 90 people, 90% are keeping quiet. But they run your body, they run your mind, they run your diseases, they run your disease mitigation, and so on. Now, these are all the microbes that are sitting over you, inside you, and everywhere inside you. Now, we said, if this is going to be the case, should we not be exploiting this? Should we not be looking at it? So we went into a deep dive. Our team huddled together, put together a process wherein we were the creators for the first respiratory platform under our company, which is Live Biotherapeutics, wherein we can sense, we can make that bug which is inside. Maybe it is already intelligent, but since we people do not know what it is doing inside, or we do not know what language it is speaking inside, we had to engineer its circuits, we need to engineer its sensors, we need to engineer its abilities to see whether it can talk to us from inside. So which means this is a classical sci-fi. So you put a bug in, you engineer it, you send it across, it will survive in hypoxic environment, it will survive in oxygen environment, it will send you some messages that you can pick in this side, which means it opens up two possibilities. You are talking about a diagnostic system. So imagine a situation where there is a cancer that is just growing. And many times in our families, we have heard this, right? We say, oh, you should have come a bit early. Now it's all too late. So we can't do much about it. Now, imagine there is a bug which tells you, man, there is one cell which is getting to be rogue. Can you just look into it? That is the type of technology that is going to go on. So if you're talking about people living up to 150 years, then this is exactly how this world is going to go through. Modern technologies, cutting edge technologies that will come. Now, I would like to also tell Mrutyunjay and the rest of the team here, I wish I could hear another person from India or running another company saying that, hey, you know what, we are also doing this. In fact, I have flagged to DBT that live biotherapeutics is not even part of their biopharma policy. So just imagine how backward we are in terms of policy writing. So that is where, you know, India has to buck up, and industry will have to come in and tell both sides, please, gentlemen and ladies, please ensure that you are able to move it to the next stage. Now, why did I bring up this live biotherapeutic segment here? I brought this because I want students to think of such revolutionary ideas. How can you make personalized medicine? How can you make detections more personalized? How can you make things much better? I normally don't wear watches of, which are doing this data, but I wanted to show you today. I mean, this is a type of watch that you can see has got data that tells you about everything that goes on around you. And there's going to be times when the microbe inside will be talking to these watches. These watches in turn will be talking to you through detection systems. And all these systems are going to be built not by me, but by you guys who are sitting there. The 200 odd people who are there are the people who are going to be now building all this. I may or may not be around to see all this happening, but at your lifetime, you're going to be wearing these watches. You're going to be wearing these things to make things so beautiful that you will have a control over how you live. What would you like to live as? What happens when you eat something? What happens when you burn something? What happens when you get disease? What happens on how you should be treating yourself? Now, this is the level of dream. This is the level of heights the technologies can go. And remember, all these are well within your reach, well within your own ability and your capabilities in Orissa. There is nothing you need to go out and search because all these are available to you. All that you need to do is to build these intelligent systems and platforms and take it up. Not only that, as you know, DNA stores information. You're going to have a storage that is going to be used by this. Google, most of the major companies are all doing this, coupled with AI, armed with the ability to put this data in a form that can be machine learned and understood. Deep learning has gone into a completely different gambit as a whole. So I would say that students should definitely look at such path-breaking ideas, then reach out to industry leaders who are doing this to be their mentors and see how we can all join this effort together to build some world-class ideas out of this place. So that, some of us who are very lonely here doing things that, only we understand and not very many people even come to understand it. It'll be nice to have a cohort of people coming in, lots of people who can come in and say, yeah, come, let's all work together and make this happen. This hub and spoke is what is the model that we should be building, where Orissa as a whole, as a state, where Bhubaneswar can be the hub and the spoke should go to all other places where, you can try to get this done here. Now, I can go on with several technologies, intermediates that we need for our country, lots of self-reliance that we need, the how within a very short time our startup ecosystem has really gone up thanks to the Prime Minister and his team. Last week was another great information that we had 26 billion for the first quarter, which has come into startups alone. Now, these are all happier times, guys. Opportunities just come once. It's like, that spark that comes in, you don't capitalize on it, then you will be regretting the rest of your life. So just simply focus on that. Keep your eyes and ears open. Be sensitive to new things that are happening and interact. And then you will have to have great alliances to take this forward. So I think your conference is doing exactly that, creating that spark, creating that vision where all of you can get together and probably forming that alliance. I'm sure, I don't know the 200 faces that are on the other side today because my Zoom platform doesn't give me all that facility as yet, but I'm sure you all have had a face to understand how I am still feeling pretty young and excited that, I'm winning competitions. I'm putting myself through the grind to see where I am in terms of my ideas, etc. So I would like to compete with you guys. I would like to compete and take, challenges from you. So this is how I have shaped my science, all the time competing, all the time on being the edge of the seat. So I would like to formally thank the organizers for giving me this opportunity. I know I have about eight minutes. I had asked Mrityunjay if I could stop a few minutes earlier, because I don't want this to be a monologue of sorts. We'll be happy to take questions. Srinivas, if you can help us in, taking some questions and asking me. If I know the answers, I will tell you. If not, I'll tell you I don't know. So I'll be very happy to do that. I wish all of you the best and your conference the best. And Mrityunjay, keep rocking. You're doing a wonderful job along with your team and all your advisors and well-wishers. Thank you very much.
Thank you, sir. Thank you.
Thank you so much, sir. It was a great talk from you, Dr. Murali, and you spoke about adopting new technologies such as mRNA technology, hub and spoke model, and a great initiation by ABLE and other agencies for giving a wonderful startup ecosystem. And I'm sure many of our startup companies, startup entrepreneurs, all those people definitely make use of these platforms and do better for the country and for themselves. So, sir, before we go to other questions, I have one question: how we can encourage young budding minds into this startup ecosystem?
See, basically, startup is, we have to be very careful about this, Srinivas. I agree with you in the sense that, if somebody should have an idea, and he should also feel that the idea is really worth taking it to the next stage. Even before he actually plunges headlong into it, he should talk to people. Is it scalable? Is it something that can be taken up? Is this something that can be moved? How can we take it to the next stage, etc.? It's not just because there's some money available, or it's just that nice to be fashionable, etc. That's not what we want. We want to be sure that this idea is a very scalable idea. It will have a global. See, don't invent a problem where there is no need for it. Suppose there's an old saying, right? If there is no problem, don't try to fix it. You know what I'm saying? So don't try to fix something which is not there. So look at the problem and try to see what can it fix, and dream big. Yeah, really, really dream big. Today, we can get that sort of money from different places. So dream big. Don't be limited by your middle class. I'm a middle class person today, but I dream of billion-dollar businesses, million-dollar businesses. With my money, I can probably buy a Maruti 800, but that doesn't mean anything to me. I can build value for customers to that extent. So that should be the type of motive you should be looking at.
Thank you, sir. Students, you have any questions? Faculty members, any questions? Please go ahead.
You have another 3-4 minutes, so you are most welcome. Otherwise, if you don't have questions also, you can always write to me, Srinivas. There's nothing wrong in that. Mrityunjay knows how to get me there.
No, I'm sure Mrityunjay sir has a question.
He's one person whom I can't hide from. That's the other problem I have.
Thank you. So nice of you, sir. No, I think, sir, can you, because the students are here, faculty are here, I know what we discussed about ABLE center, something like that. So what is going to happen in this?
In January, I think Mrityunjay.
So what is going to happen? Because the Srinivas Patnaik should know and the faculty should know what is going to happen. How we are going to build that platform here.
Yeah, see, the idea is that we have several platforms of this kind in Bangalore, right? So the idea is that some of the times you don't have to reinvent the wheel. So the whole purpose of getting into Bhubaneswar will be to see that there is an industry outpost which is listening, and that outpost should soon transform itself into a full-fledged center, so that any ideas somebody comes up, the earlier he comes up, the earlier he understands if that idea will be going through, the earlier he gets funding, the earlier there is an alliance, the better it is. So that is the disconnect that happens in most parts of the country, because they are all living in silos in their own little hamlet. where we need to do is to provide the stage where, they understand the world is in their hands. So that is exactly what ABLE did during BEST program. And that's exactly what we would like to see happening more in that place. Because right now you are taking industry on a need basis, meaning that they come, they talk, they interact, they go away. But you shouldn't allow them to go away. Literally, physically, virtually, they must be available to you all the time. So that's the platform that this is going to do.
But also they will come and, there will be a regular program where the industry will come and talk to our people also, right, sir?
Yes, I think so. Because you see, industry will automatically gravitate, Mrityunjay, where there is talent, where there is opportunity, where there is some way of doing things. Don't worry about that at all. Industry will do that. They are known for that. You don't have to even, request them with folded hands, because they will just simply come there. That is their job. They have to go and seek opportunities.
Good, sir. Good. Thank you. I think that's what we are expecting, because we don't have much of industries here, biotech industries, except mining. So I think with ABLE, we want ABLE should enable this now.
We will be happy to do that.
Thank you, sir. Thank you.
Thank you very much. I think my time is almost done, Mrityunjay. I think you should carry on, and I still have to travel.
Thank you. I just messaged the other speaker. Anil Kaul, right? Murli sir? Anil Kaul, who was the director of IMTech Chandigarh. Then he again moved back to Johnson in Belgium.
Oh, wonderful. See, we could have met, we could have spoken. I'm not sure
So he's the one who discovered it beta-quilin for the MTB, MDR TB.
I see. Wonderful, wonderful. So nice, so nice.
So he's going to speak. I just called him. He said, oh my God, I forgot. He was sleeping. He said, give me 5-7 minutes. So he's getting ready.
Where he's in Belgium?
He's in Belgium, and he's also a great orator. I think bring a lot of interesting science, but he builds products out of it. A lot of trials, things have been happening.
Wonderful, wonderful. I will leave some time for you gentlemen to refract, because as you know, Mrityunjay, I have to have a 5-hour car ride from here. So I'll have to start moving, and I have a series of AGMs and others in the evening. So I would like to wish all of you the best, and my good wishes to all of you.
Thank you so much, sir.
Thank you, sir. Thank you. Thank you very much.
Thank you. So
dear students, I think, when we are calling such people, you have to ask questions. There is not a single question people are asking, not a good idea. So right now, Anil Kaul is going to come. Please see that, because he's going to bring a lot of interesting science, then product, and he's from London School of Tropical Medicine, and then also from Johnson & Johnson. I think you have to impress these people, right? All the best. Keep on making those questions, prepare those questions, smart ones.
More questions, more industry connect. So better be on the field. So thank you so much. I think, shall we wait for a few minutes, sir?
Yeah, so he's coming. I just called him, and then he got up. He said, oh, Murthy, he thought it's 11 o'clock. I said, no, get ready. So he's coming in 5 minutes.
Okay, sir. So is that okay, sir? Yeah, okay.
No, you can run, you can start anything. You have anything to talk, you can talk.
Gopal, sir, if you don't mind, can you please run that video again? So that
Sure. We'll do that.
And see, somebody should allow Anil Kaul ji.
Yeah. Gopal, sir, make sure that, once he enters, we allow.
Yes, we'll do.
So he has just sent an email. It's coming. It's delayed by 15 minutes. Okay. So why there are so few students only? 222 out of that, maybe there will be 20 hours.
Others are on the YouTube live.
Okay.
It's because of the Zoom link limitation.
Okay. But it's 300, right?
Rest all batches are on the YouTube live.
Okay. Okay.
So Gopal sir, please ask the 7th semester students also. I am dead to join in this Zoom link.
So it's on the YouTube live. So let's not
YouTube
discuss
live.
on.
Yeah.
So let's not discuss here. We are on live, right? It's the social sites.
Yes. Sir.
Good. I think we should wait. I think Dr. Call is there. Already arrived.
Yes, sir.
Anil sir, good morning. Sorry.
Good morning, Mr. Sorry, I'm.
No. Sorry. I know it's too early in Belgium.
It's okay. It's okay. Not that early, but I was I was trying to bring my kid to the school.
Okay. So yeah, I think Pratnaik, you can start now.
Yeah. Good morning, Professor Anil Kaul. I was just checking the time. I think it's 7:40 in the morning. So sorry we woke you up. But so to start the talk, I think before that I will just give a small introduction of Professor Anil Kaul. Currently, he is the Vice President, Global Health Discovery Research at Johnson & Johnson. He's currently Vice President. He's heading the Global Public Health Discovery Partnership. And he holds a professor position in translational research at London School of Hygiene and Tropical Medicine, London, where he set up a laboratory of translational discovery research. He also serves on Scientific Advisory Committee or Council of Scientific and Industrial Research, CSIR, Government of India. He was also director of India's premier public research lab, CSIR IMTech. His biggest contribution in the discovery and clinical development of bedaquiline. It is the first drug to be approved for the treatment of drug-resistant tuberculosis in last 45 years, and it is WHO's list of essential medicine. It's a very, very huge discovery for the humankind. For the discovery of bedaquiline, the Government of India awarded the Sun Pharma Research Award in 2017, and in 2020, he was awarded the Hero of Chemistry by the American Chemical Society, and he is that was awarded 2016 Prix Gallen French for the discovery of bedaquiline. He published several research articles in Nature, Science, among many other international journals, and he holds several international patents. In a nutshell, he works across 3 continents, America, Europe, Asia, and passionate about innovation, science, and cost-effective R&D strategies in public health. He's a pioneer in science, and he has a great vision to bring biotechnology industries in reality. So welcome once again, Professor Kaul. We will look forward to your deliberations. Thank you.
Can you hear me?
Yes, sir. You can.
Perfect. So, good morning, everybody. It's so wonderful being here, talking to you all, and my apologies for the delay in getting to you. So as Professor Patnaik said, that my name is Anil Kaul. I'm a scientist by training, and I have been fortunate, one of the very fortunate persons to visit KIIT, to visit School of Biotechnology, and to be there. It was somewhere in I don't know, it was something 2017 or 2018. And it was so wonderful being there to see all the amazing work which Professor Saur and everybody else in KIIT is doing, including the TBI setup, what you have, the biotechnology incubation, what you have, and all the startups which you guys are trying to pull through. So it's very important, not only from the science perspective, but also very important from the innovation perspective, as you can see that how basically science is driven by a lot by the industrial setup. That means if you look at the global science funding, and let's take US, a lot of US science funding is driven by industrial funding. So almost 70 to 80% of funding comes from the industry. And this is somewhat in contrast to what we see in India. A lot of science is driven by funding, is driven by the government. So we need to have, of course, the biggest challenge right now is to have more industrial funding, more industrial opportunities, more industrial scope in innovation, because that brings up the scale of innovation heavily across across any country, like Israel, for that matter. You see a lot of science is funded by industry, and the same thing in all the developed countries. South Korea. South Korea, I think more than 90% of the funding for science comes from the industry. So that reversal is basically part of what we need to do, that we need to not always rely on the government sources for funding and doing innovation in science, but we should really look for the private setup, the private industry, what they are trying to do, and how they can fund the science, both basic research as well as what we call public health research. So I will try to give you in next 30 minutes, I will try to give you a glimpse of how we are able to do it at J&J. A basic outlook in terms of not exactly framing in the same way which I have framed it here, but try to give you a glimpse of collaborations and partnerships and alliances which are important for, especially in the public health, or for that matter in any field, whether it's medtech, whether it is oncology, or whether it is very other forms of science which are heavily funded at the industry level. So let me share some slides with you, and my apologies, some of the slides you will see as a repetition, but I will try to bring in new perspective in terms of yeah. Can you see my slides?
Yes, we can.
Yes, sir.
Wonderful. Okay, once again, thanks for this invite. It's great to be on this Biotech Industry Conclave. And it's really wonderful that when you chose the topic Alliance and Deputation, empowering the backbone of the biochemistry. So from that perspective, these alliances are very, very important in terms of innovation. And we have seen it with COVID. We have seen how important it is for us to be partners with the government, partners with the public, partners with the industry for a very common cause. So my theme today will basically serve the main idea of how important these partnerships are. So I think some of you may have seen it last time when I spoke, but I still want to tell you, keep on emphasizing how critical innovation is and how important innovation is to think differently. It's not to think and use the same idea which everybody else has used. So thinking differently is very, very important. And that is where you have the disruptive innovation. And it has happened in HIV. As some of you may know, that HIV was a death threat in when we say in 1980s, late 80s and early 90s. HIV people used to die. Means it was within 6 months. Now nobody from HIV will be dying because you have good medications. And the same thing with the hepatitis C. It was really, really terrible to have both these diseases, but now these diseases are manageable. It's not that we have cure for them. We cannot cure HIV still till now, but we can manage the disease effectively. The same thing in terms of immune oncology, where you have immunology has really drastically changed the field of oncology. It's really amazing understanding the immune system how important it has driven the field of oncology, the field of other diseases, whether it is directed T-cell therapies, or whether it is CAR T therapies, or whether it is any form of immunology. We have a touch of oncology, we have a touch of immune system coming into play. And the vaccines, we all know, whether it's the mRNA vaccines, and whether for COVID, or whether it is the classical vaccines for streptococcus or HPV, they have changed and saved the human race from pandemics, and also increased the decrease the childhood mortality for several of these diseases. MedTech is a big, huge innovation where you have the AI, you have the ML, and future for all the surgery. We have a big, huge surgery group in J&J. It's one of the biggest medical devices and devices units across the world. So, and when you talk to those people, they say that the surgery has a lot of robotic component which is going to come into play in next 5 to 10 years. So a lot of surgery will be assisted by robots, and it will be minimally invasive. That means you won't be really needing to cut through the whole human body in order to do a surgical procedure. So the same thing we have seen at J&J, that how we are trying to incorporate digital health or data sciences. So it's a big, huge push right now in getting data sciences in the company and across the world, because we have seen how important it was to have data analytics. Not during the COVID times. I can give you example of COVID, where we use the data sciences to identify the trial sites, to look at the sites which had maximal chances of enrolling the patients which had COVID, or where the COVID was going to evolve in the next 1 or 2 months, and what kind of isolates were there and things like that. So this was all driven by modeling, big data analytics, artificial intelligence, and all those tools. So definitely a big, huge push in terms of innovation is basically the big data. And finally, I think the key is, I have spoken it earlier, that the mRNA platforms, how a technology which was no one was using and no one was believing a few years ago, and that mRNA is something which we can really use to deliver and develop vaccines. Now you see the mRNA, not only for COVID, now you see the mRNA platforms basically changing the whole landscape of COVID, and also have a huge potential in terms of other infectious diseases, as well as for oncology. So can you use therapeutic vaccines using mRNA for treatment of certain forms of cancers? So some of the pictures you see are basically from the Biotech, from Kathleen Carico. She's the one who basically has been working 20 years on trying to deliver these mRNA. It's a very simple concept, but the challenge was the delivery of mRNA. How do you deliver? Because as you all know, mRNA is such a short-lived molecule. So the key was to stabilize it and deliver it. And that 20 years of work in the basic science really pushed the field heavily in terms of developing those products. So you need a very strong element of basic science to develop the product, and that's what this all mRNA research, starting at University of Pennsylvania and now going to BioNTech, Pfizer, and other companies like Moderna and others. So indeed, a lot of basic work, and that gets translated into science. So how do we I'm just talking about a very small unit of J&J, which is public health. We call it GPH. So why do we do global public health? Because it's not always about money. It's also important to look at and what impact you have in the lives of people. So these are what we call non-commercial programs, and that's why you see the focus areas have always been tuberculosis. Leprosy is still a big, huge issue across the world. We have not been able to eradicate leprosy. It's one of the oldest human diseases. HIV, as I said, that needs a cure platform. COVID, you all know the vaccines. As you know, we have a COVID vaccine based on the adenovirus platform, Ebola vaccine, and also one of the very transformational HIV vaccines. Intestinal worms is a big, huge challenge in a number of countries in the world. And then we have flaviviruses, dengue, and AMR. So apart from these infectious diseases, there are big challenges in the developing world is the mental health. And it's underscored a lot of times, but mental health is a really, really important aspect. I will give you some examples what we are trying to do. And then you have essential surgery and maternal health. Essential surgery is basically a lot of surgical procedures across the world right now are happening not in an optimal conditions or optimal infrastructure. So how do we strengthen that essential surgery infrastructure? And always the maternal health, if you look at the cases of neonatal sepsis, so it can tell you that how much important is the health of a child, newborn child. So once again, in terms of the technologies, that's really the disease form. And if you look at the technology form, what are the key drivers? What we are doing, or my group is trying to do, is looking at digital health. As I said, that this digital health and digital transformation has been very, very helpful in getting the fast regulatory approvals, in recruiting the patients, in identifying the right patient population. So that's very important. And also being able to, like Ebola, we used an iris scan platform to do clinical studies in Africa, which nobody has ever done. And I can tell you this was one of the best things in terms of being able to recruit these patients, and also being able to follow up using the iris scans, and eventually the vaccine was very effective and the trial was very well done. The same thing, data sciences and long-acting platform. The question is, long-acting means that if you need an injection once every month, can you give, instead of every month, can you give it every 6 months? Can you give once a year kind of an injection? So that's what we call long-acting molecules. And diagnostics, of course, is a big, huge component of all what we are trying to do. These are some of the transformative outcomes. When we say, so you see on this slide, you have 4 pillars. You have transformative outcomes, you have emerging impacts, you have next generation science, you have differentiating capabilities. So the transformating outcomes is basically looking at the outcome of Betaclin, looking at the outcome of a drug which has a huge impact in the world right now. I will speak a little bit more about it because this is my favorite baby kind of a thing, but we are looking at how do we deliver therapies, especially long-acting therapies. We know the challenge in some of the low and middle income countries is the mental health, which has been neglected. So there is where we have risperidone and paliperidone as long-acting injectables to treat a disease like schizophrenia and things like that. I talked about vaccine platform. I talked about emerging impact, especially how do we eradicate the leprosy from the world? we have a very nice program on dengue. I know there was a huge problem in UP recently around dengue, and we are looking at dengue therapeutics. How do we basically have a small molecule which can be used in times of dengue pandemics or epidemic, and or for treatment of dengue symptoms or killing dengue virus. The same thing, we have safe birth, which is very, very important, a team looking at fistulas and postpartum hemorrhage and during, as I said, maternal health and soil transmitted elements. So next generation is still working on science, is working on developing TB regimens which can kill TB in less than 3 months. And we have a big, huge program on COVID. A vaccine, you already know, everybody knows, but we also have a program targeting small molecules just to make better, effective therapies for COVID. And AMR, I will talk a little bit more about AMR. So doing all this, what I'm trying to say, doing all this needs partnerships. We can't do it alone. It's almost impossible to do everything on your own. So what we try to do, when I say differentiating capabilities, we have platforms where we try to build those partnerships. So one of the platforms which we have developed is basically what we call resource limited setting operational model, or African clinical trial setup. So where we develop how to do trials in Africa in much better way, because otherwise these patients are missed. So we don't have data coming from one of the biggest regions in the world. And then the second thing is what I will talk a little bit more is the global public health discovery centers. What do they mean? Why we are doing it? What's the aim? And then we, as I said, that we have Center for Pandemic. We are building. We don't have it right now. We are building Center for Pandemic Preparedness and Response, and a lot of other external financing and unique partnership models. So a lot of models in terms of development. So when we look at the discovery, the key challenges right now in terms of pandemic, I talked about emerging science, is still TB, is AMR. I will talk a little bit more. What does it AMR mean? Dengue and flaviviruses as the climate changes, as we have issues with the world warming up, we see a lot of flare in dengue, especially in Southeast Asia. Singapore had one of the worst dengue epidemics recently, and the flaviviruses are also a big, huge challenge. So you see those viruses which were previously subdued are emerging heavily across the world. And the same thing is that we all know how COVID emerged and what we are trying to do. So because the talk is more focused on the public health, I try to give you insight on what we call global public health satellite discovery centers. So what our approach is, as I said, that our approach is to basically partner with the major public health research institutions across the world. So what we do is that we go and try to set up labs at these institutions. And the aim is to look for early stage and translational discovery. So because we know there's a lot of talent externally, a lot of passion externally, and there is where we try to say, okay, let's partner, let's set up these labs. These will be J&J run labs and focused initially on AMR, TB, pandemic preparedness. Of course, why us? I won't go in detail, but the aim is, what is the aim? Amplifying our impact. So the aim of each satellite discovery center will be very specific, will be very project driven, will be very, very something which we will monitor every day in terms of the science and the outcome. So, and the aim is to advance the new therapies for antimicrobial research, next generation TB, pandemic preparedness, and things which normally nobody takes care of. we want to be close to the innovation. Innovation is key. So we want to be close to the innovation and public health institutions like KIIT, like other institutions across the world are basically driving the discovery and innovation. And we want to be close to those ecosystems. And as you see that help J&J scientists collaborate with the best talent in the world, contribute to the early discovery, biomarker research, and translational medicine. So how is the operating model? So operating model works like this, that we have this, the J&J providing scientific and translational expertise, because that's where our expertise. We know how to develop products. We know how to build products. We know how to do clinical trials. We know how to do clinical development. We know how to do preclinical development. So, and we bring in the seed investment, and the partnering institution provides the scientific talent, infrastructure, and research ecosystem. And then we try to rope in other contributors, basically academic partners, government bodies, and other funding agencies. So it becomes an integrated effort to drive the global public health innovation. And just to show you a glimpse, none of this is up in stone, but only thing which is defined is a lab in London School, where I also hold an honorary position, where we are looking for TB system biology, but we have plans for South Africa, Singapore, India, Germany, but these are all plans. Right now, nothing has been finalized as for now. So what we have done is at London School, we are trying to set up this TB system biology and metabolomics group to accelerate TB drug development through innovative translational science. So I will talk a little bit more on TB, but the aim is to bring science to the forefront as we talk about these public health institutions. So metabolomics is a platform which you can see has a huge impact on the drug development. And that's what we want to do. And that's what we will be doing at this partnering institution, in order to, with the overall aim is not to do just science for the sake of science, but to drive the innovation. So I talked about AMR. I will quickly, briefly talk to you why AMR is important. So AMR is what we call super infections. And AMR is going to be one of the major global public health threats in next 10 years. And now it's already, according to WHO, one of the top 10 global public healths right now. But in the next 10, 15, 20 years, we will have 10 million deaths every year by 2050. So that's a huge number if we don't take care of AMR. One person every 3 seconds dying due to what we call super resistant bacteria. And it will have a huge impact in the globe, what we have already seen in terms of the economic value. Most of the challenges, the AMR will have an impact in low middle income countries. And you can see those numbers from WHO is listed here, that how the impact will be across Asia, Africa, and the rest of the world. So these are some of the critical pathogens. I won't go into detail, but none of you must have heard about Acinetobacter baumannii or very few of you as a pathogen. I also didn't know about it very much before we looked into the details. Apparently, Acinetobacter baumannii is spreading across the world at such an alarming rate, especially the resistant forms, the carbapenem resistant. And it causes a lot of blood infections. It causes a lot of pneumonia, hospital-acquired pneumonia or ventilator-acquired pneumonia. And you can see the global patient burden. So I'm talking about MDR Acinetobacter or MDR infections due to the gram-negative pathogens. You can see how much is the global burden. So a lot of neonatal sepsis is one of the biggest challenges the world faces right now. And it's emerging out. It's not only the bacterial sepsis, it's also the viral sepsis, but definitely a big, huge challenge. Just to give you an example of how the big challenge of AMR is going to be. You look at the column 5. This is the report which came in August 2021. This is report as old as 1 month. And look at the column 5. You look at the red box. You see MDR Acinetobacter baumannii, and you see the level of resistance to the drugs it has. It's almost 90% resistant to most of these drugs. And when we looked at what is the impact of this resistance, and when we looked at a WHO GLASS report from 2018 in Africa, it's a data from South Africa on the right-hand side of the picture. So on the x-axis is the proportion of patients with resistant isolates, which they identified of Acinetobacter species in a bloodstream infection patients. And on y-axis is the type of the antibiotic which what the patient was resistant to. You can see they used amikacin. It was almost 80%, 75% resistant. Gentamicin was almost one of the very old bad drugs, which almost make you lose your hearing. Imipenem, very nice carbapenem, almost 80%. Meropenem, almost 80%. So most of the drugs which are routinely used to treat these bloodstream infections, you had resistance. And the same thing is that with the tigecycline. It was the only one, but is not approved for Acinetobacter, which was still okay. So you can see the challenge that we are losing drugs at a very fast rate, and there is no one, because it's antibiotics is not the failure for R&D, it's a market failure basically. So how does it look now, and how will it look in next 20 years? In the MDR gram-negative bloodstream infections, and this is just a data from low middle income countries. You have high income countries, you have middle income countries, and low and middle income countries. You have global patient burden of 3.7 million right now, and it's going to be 5.5 million globally. And most of the burden of these very serious pathogens will be in low middle income countries. Out of that, the biggest challenge is not E. coli. The biggest challenge will not be Klebsiella, hopefully not Klebsiella, but the biggest challenge is Acinetobacter. Though right now it's only 12 to 9%, but MDR cases are increasing, and it will definitely improve. Just to show you data from the Indian Council of Medical Research, which was between June and August 23 during the COVID times. So all these COVID patients, when they went to the hospital, they got hospitalized in the ICUs. The first infection they get is from Klebsiella. So those patients who got infected and those who did not get infected, those who got infected, they had 56 to 60% mortality because of the infection versus only 10% which did not get infected. So you can see that how much these MDR things or drugs increase the mortality in COVID patients and hospitalized setting. And as you can see, most of these patients got a Klebsiella, which was 30%, followed by Acinetobacter baumannii, which was 21%. And that is okay. You got these infections, but the challenge was that the moment you got these infections, there was nothing left to treat these infections, because you can see the level of resistance to carbapenems, which are used to treat these infections, was 92% for Acinetobacter and 78%. So most of these patients had to die. That's why you had a very high mortality rate, because there was nothing left to treat these patients. And apart from they already had COVID. So definitely a big challenge. So when I say it's all a challenge, that's why we come to discovery. That's why we come to public-private partnerships, that we have to discover new innovations. We have to discover new drugs. I'm not just talking about AMR, it's about every human condition, every human health, and these pathogens basically want to disrupt and want to develop. So let's go to TB now. TB is classic because it's close to my heart. We know 10 million people fell ill within TB in 2019. So that's the number. So 1.5 million deaths in 2019, and it's going to increase. Its projections are around between 1.8 to 2 million deaths because of the COVID impact on TB. Number one cause of the AMR infections, 500,000 developed MDR TB infections. So really, if everything goes like this, we will have 32 million deaths, much more by COVID by 2050, and a lot of economic loss. So this is an old slide just to show that how we discovered the Betaclin, just to show you that the innovation and the science we do in lab really translates into products, really translates into drugs. So we discovered this molecule, which was a very small molecule at that time. It was called TMC207. It's called Betaclin. At that time, we discovered this enzyme as an ATP synthase for Betaclin with the IC50 of almost 2.5 nanomolar. Very nice specificity in terms of the inhibition. So nobody believed that, and the target can be ATP synthase. Nobody thought that this should be the target for the enzyme, because everybody thought that you cannot block ATP synthase because it's so ubiquitously present. But over a period of time, we somehow managed to pull the program through and do the right science, build the confidence in the molecule, and eventually develop it. So this is the cryo-EM structure, which was recently published. I will show. I like this picture very much because it shows you at the molecular level how the ATP synthase, which is a very complex machinery, and how Betaclin basically binds to this molecule and blocks its function. And ATP synthase, as we know, is basically an enzyme involved in the synthesis of ATP. So I will play this video. This is the C subunit of the enzyme. And you can see this Betaclin drug coming in and blocking the interaction between E and C subunit. And the moment you block this interaction, you block the rotation of the enzyme. And this rotation of the enzyme eventually leads to what we call blockage of ATP. And we know that ATP is such a fundamental source of energy for TB or for any other cell. But we know that these drugs block ATP synthase very specifically. So they are very specific to mycobacteria, don't have an impact on any other bacteria, and also very highly selective to the human mitochondrial ATP synthase. So this was a study done by a group in Canada, and they published this cryo-EM structure of ATP synthase for the first time with the drug Betaclin recently in 2021. So yeah, I will just tell you that where we stand today. This is the impact of the innovation, that that the drug is available in more than 143 countries since its launch, has been registered in 66 countries. And that means it has the regulatory approval, and it's part of WHO's list of essential medicines. It's part of WHO guidelines, and it is being distributed across the world. I think it's not only adult indication. What recently has happened with this particular molecule is the approval in pediatric setting. So it has gotten approval for pediatrics. So 250,000 of kids die every year because of MDR TB. And you can see that India has recently in 2020 approved it for pediatric trial and pediatric indication. And there are a lot of other countries where we are still looking at approvals for kids between the age group of 5 and 12 years. So yeah, this was approved. I think I will skip this slides. So we are, as I said, that we are still working on developing new regimens. So my goal in life is that can we develop a drug for TB which kills TB in 1 month, or which cures a patient in 1 month. And right now, TB still needs 10 to 12 pills every day, especially the MDR TB. Can we have a pill which is just a single pill? So we are developing the science, we are developing those systems, we are developing those what we call TPPs in order to drive that innovation. So it's the same thing, like we have energy as a source of killing TB. So we have ATP synthase, and we know that we can switch off the energy of the bacterium and by using Betaclin, but we have other switches. That's why when I said the London School metabolomics, the metabolomics impact will have a huge impact on what we are trying to do here in order to understand which other switches are there so that we can supply the energy of the TB bacterium and shut down what we call the power grid. So this is a slide interesting from a personal perspective. This is a slide where we took this picture in South Africa, where we went once. I think it was in 2016 or 2017. When we were there, we looked at one of the hospitals which was treating MDR and XDR TB patients. And what was very surprising when we went to the pharmacy of the hospital was to see these BL boxes. And when we asked the person, what are these BL boxes? They said it is Betaclin. So it was very satisfying to see the science, what we do in the labs, getting translated into the products. I think I won't go into this in detail. I think it's very important for this young group to understand what drives innovation. We keep on talking about innovation. I think it's very important to have the internal expertise. That's very, very important. You need to spend years and years to develop that expertise. The failures will be there, but we need that. And as I have demonstrated here, we need external networks. You need that networks to develop that expertise as well as to improve your science. I think entrepreneurship, we all know, is critical. Operational excellence, and of course, your heart and soul in the program is, you need the belief that what is happening is really true. And of course, the leadership and talent is critical. I think this gives me hope that in the Indian context, there's a lot of potential and a lot is already happening. We don't see it visibly at times, but I can tell you, I've been part of the system. There's a lot of progress which is happening in terms of increasing the innovation scale. And I can tell you that it can improve further if there is more participation from industry, if there is more industrial R&D happening, if there is more scope of developing those startups and bringing them up to the scale and driving that innovation. So there's a lot which still needs to be done, and we have seen across the world how this can be done, and we know that this India is on the cusp of breaking through the first 20 countries which are most innovative. Finally, I think this is what I call, I don't know, there's a movie on Netflix, if somebody has seen on what they call it Inspirational 4. So these are the 4 people who have been selected that to go in the outer space. So these are the random 4 people like you and me, and this was unthinkable. This shows you what we can do just by the power of the vision, that nobody else thought that a space mission can be a private sector enterprise, can be run by individuals who have no experience of. Everybody thought that nobody can do what NASA is doing, but SpaceX showed that they can do a job and very professionally. And this is what we call loonshot strategy, that you have the biggest dream, It may be a crazy idea. It may be an idea which nobody wants to believe. But if you have a dream, I think it always comes true. It just needs the power of resilience, the power of pushing the boundaries, and power of being honest around that loon shot. SpaceX, you should follow up the stories. It's very interesting if you are interested in the space technology. And they had almost 6 or 7 failures. They were almost completely bankrupt. And now, since 2016 and 2017, you can see how many failures they had. And now they have graphed their technology. And whether it's the right thing to do or not the right thing to, that's not the question, because a lot of people criticize them. Look, we have much more problems on Earth, and you are talking about space. But that's a separate argument. That's a philosophical argument. What the argument is here, that few people could really muscle together, bring together the right kind of leadership, bring together the right kind of people, and yeah, they made possible which nobody thought that this will be possible. So that's the end of the presentation. I would like to thank you for being with me and bearing with my delay. And I will back to Srinivas, back to you.
Thank you so much, Professor Anil Kaul. It was a wonderful talk, and you can see the chat box. It is flooding with questions. So because we are running a little late, I'll just ask one question. Rest of the questions maybe I'll forward it to you later. In the time you can just send it reply. So one of the questions, the discovery of beta-quilin, to get it listed on WHO's list of essential medicines, what were the hurdles that came on the way?
That's a very nice question. That's a very fundamental question. I think, to be honest, I can give you the whole story, emotional story. I can start crying. What were the challenges? I think there were a lot of challenges. The biggest challenge was we had a clinical trial which didn't work very good, and the project was almost to be stopped. So, and a lot of people don't believe. At that time, not many people believed that this molecule will be successful, and there were a lot of people who didn't want it to be successful because, yeah, you can imagine it's not financially attractive to develop a TB drug. So I think, but there were a lot of support. A lot of people at the same time supported and helped and put in that investment to bring one of the most essential medications for TB right now. And I think, yeah, there have been definitely a lot of scientific challenges, operational challenges. It's not me alone who did it. There have been hundreds of people who have been involved in this project. I'm just speaking on behalf of all those people who were part of the whole story. So there were a lot of people in clinical development and pharmacology, and still there are a lot of hundreds of people who are working to make sure that we don't develop the resistance, the drug is effectively used, the drug is available across the world. The drug is, because you have to imagine that somebody in the remote corner of the world needs the drug, and it has to reach them. So how do you make that supply chain component of delivering these medicines freely available or be present? So a lot of challenges. It's not, discovery is one part and development is one part. The product development is how do you bring finally it to the patient? That's much bigger challenge for some of these public health indications, because it's not, you don't have the sales force, and you don't have this, you don't have that, you don't have the right, you cannot employ thousands of people. You just need to work through channels and be most cost effective in bringing it to the bedside of a patient.
True, sir. The similar kind of trend we are seeing right now in case of this COVID vaccines and many other things also simultaneously. So, sir, the chat box is flooding with questions. So I think looking at the constraint of the time, I think we have to say a great thank you for Professor Anil Kaul, and thank you very much, sir, for your deliberations and your time. Thank you so much. And we look forward to again invite you to School of Biotechnology sometime, and we'll definitely listen to you in the offline mode.
Definitely, and once again, thank you, everybody. I think the School of Biotechnology is an amazing place, and the people you create. I have seen at so many platforms the students you create. The kind of training you guys give is really amazing. So, and these are next generation leaders. So, so that your alumni is, and the pool is growing really good. And the training, the openness and the transparency and the opportunities you create for these students, I really feel that you guys are doing a really wonderful job. So congratulations to the whole school for giving aspirations, hopes, and dreams to all these people. That's what matters in the life. Do we have the hope? And do we have a dream? And if you can give it to them, I think nothing matters more than that.
Thank you so much, sir. Thank you very much for your inspirational words. Thank you so much.
Bye-bye.
Bye, sir. So thank you very much to Professor Anil Kaul for delivering his keynote address. So now over to Dr. Rahul Modak to take it to the next session. Over to Dr. Rahul Modak. Thank you.
Thank you, Dr. Patnaik. So I welcome all the participants for the next technical session, which is molecular diagnostics, the front runners of the novel battle in the biosphere of science. Molecular diagnostics is a vast domain of healthcare that is touching our everyday life. It is an emerging field that is enabling doctors, healthcare professionals with informed decision making during treating patients. Presently, molecular diagnostics is emerging from protein-based diagnostics to DNA-based diagnostics, which is helping in rapid detection and prediction of disease progression. Importance and innovation in the field of molecular diagnostics are quintessential in the present day of life. Collaboration of different analytical techniques is crucial to optimally analyze the patients and disease, in turn creating personalized healthcare. It is important to note that molecular diagnostic tools are extended to agricultural and animal husbandry also, which is ensuring food safety for today and for future. Today, we have 3 stalwarts from industry who are pioneering these efforts in India, and they are recognized globally. We request the luminaries to share their views on the following topics. How difficult first topic is, how difficult and technically demanding is the quality control and quality assurance in case of molecular diagnostic assays? And the second question is, what will be the cost effectiveness of the pharmacogenomics industry look like in the near future? So today we have 3 speakers with us. I welcome all the 3 speakers. Mr. Ashish Dubey from Redcliffe Life Sciences, Mr. Sayantan Barat from DSS Image Tech, and Dr. Srinivasan Vedantam from MedGenome Laboratories. I'll first request Mr. Ashish Dubey, CEO and co-founder of Redcliffe Life Sciences. I will give a brief introduction about Mr. Dubey. He is the CEO and co-founder of Redcliffe Life Science. He has bachelor's degree of science from Purvanchal University and a master's degree in science from Chaudhary Charan Singh University. He holds a diploma in clinical research from ICRI. Mr. Dubey has worked as a manager life sciences at G Biosciences USA and at Sander Proteomics Private Limited, which included multifaceted roles of sales, marketing, brand establishment, and promotion of scientific training. Before taking over the roles of the head of sales and marketing, sales head and business unit head successively at Sander Proteomics. His two companies, GenePrint Life Science Private Limited and Redcliffe Life Sciences, New Delhi, unitedly catered the need of DNA-based diagnostics and research using high genomics technology. May I request Mr. Dubey to share his views about the topics of the day.
Hi,
Over
Dr.
to you,
Rahul.
sir.
Hope I'm visible.
Yes, yeah.
Yeah. If you don't mind, can I switch off the video so that I'm more
Sure.
clear?
No problem.
Thanks so much. Okay, great. So thank you so much for inviting me. And once again, it's a pleasure being here. And I think it's my second successive year with you guys. And what an energy I can see in the students and the questions, which is very relevant in the scientific discussions. I think the question which you have thrown to us is a very valid in the given time. And today, when we see in last 2 years what COVID has done to the country and to the entire universe, molecular diagnostic played a significant role, combating the problem, managing the issues, and also, providing the right and affordable diagnostics, to the needfuls. At the same time, molecular diagnostic industry has not seen the growth in last decades what we anticipated, and thanks to COVID, well, not really thanks to COVID, but thanks to COVID for the industry, that it has seen a different surge of opportunities. And I just hope that there is no other diseases like this coming to the universe. But yes, industry is now prepared, the country is prepared, and everybody who is part of this sphere and the ecosystem, they have seen how to really go scale and reach to on different heights within very short span of time. India have done a wonderful job. Companies came forward faster than ever. They delivered what was the need of the hour. And again, this is only because there was a requirement, there was a need of a scale. So every industry which is today available in biotechnology, it is all basically looking at the scale. And if you remember the 4,500 rupees per sample test, today it is 700, 400 rupees, which is 10 times lower in just one and a half year of time. That's where it has seen, the difference. And the difference is only because of the demand. So all these industry which works for the healthcare or whatever industry we talk about, it is all about demand, and molecular diagnostic is one of it. I hope that this trend continues for the research and development activities in the sphere, and we continue to kind of discover and bring down the cost for many other tests. I was hearing the talk before, the molecular tuberculosis, MTB, and many other diseases which has been discussed today. I think this is all need of the hour, and we definitely need faster and economical solutions to fight such deadly diseases in this country. That's my view, Dr. Rahul.
Thank you, Ashish, for this very interesting aspect. And you brought that very critical point that, yeah, industry is now ready. and we really need to explore more opportunities to expand our horizon for the molecular diagnostics. Now I will invite Mr. Shayantan Bharat, Business Head, Molecular Diagnostics Division, DSS ImageTech Private Limited. Mr. Bharat is currently Business Head of Molecular Diagnostics at DSS ImageTech Private Limited. He completed his MSc in Microbiology from ICMR, Enterovirus Research Center. He has experience in multiple sectors in different companies, including as the product manager in Transasia Biomedicals Limited, senior manager DGM Marketing at Merrill Life Science Private Limited, associate vice president, head of marketing at Trivitron Diagnostic Private Limited. I'll request Mr. Bharat to share his views.
Thanks so much, Dr. Rahul, and good morning to everyone. And really appreciate what you're doing for the institute at your end. So I had a few views on the entire thing, and I won't take more than 10 minutes, I hope so. So for nearly 2 decades, we have been envisaging that molecular diagnostics will be the front runner in diagnostics tests. But unfortunately, this has really not happened. The main reasons being the commercials, wherein the molecular tests are expensive. And since the walk-in pathology patients in India are not covered by insurance, doctors and as well as pathologists shy away from prescribing such expensive tests. Furthermore, the lack of specialized laboratories and personals also have contributed to the slow advent of molecular testings in India. Molecular testings have seen progress in the last few years, which promises better outcomes and accurate results. The possibility of detailed identification of tumors, mutations, antigens propels this technology further. This has also been aided by the initiation of government projects like National AIDS Control Program, which is NACO, and RNTCP, which is a revised National TB Control Program, based on molecular technology, and this has given a substantial boost to the advent of molecular diagnostics and routine diagnostics. The main technology in molecular testings is polymerase chain reaction. This continues to be the choice of assay, mainly because of the availability and comparatively instrument-dependent interpretations. PCR accounts for more than 65% of the market. Other technologies in the segment include INAT, which is isothermal nucleic acid amplification, ISH, in situ hybridization, microarrays, which we see now coming to the forefront. We see a lot of NGS, which is next-gen sequencing coming into the forefront, especially for neonatal diseases. The Indian market today is approximately 350 crore, and HIV, HBV, and HCV and the TB testing programs nearly contribute 75% of the molecular testing being carried out in India. Now coming to today's scenario, the first case that was recorded for COVID was on 27 January 2020. A 20-year-old woman was presented in the emergency ward in General Hospital in Trichur, and she had traveled in from Wuhan, China. And then by March 2020, we were witness to the national lockdowns and restrictions as the outbreak had reached our doorsteps. The nation and laboratories were really not prepared to handle molecular testings at that large scale. Even some of the big chain labs and hospitals did not have molecular testing facilities. It became a nightmare for our symptomatic patients, and even to get a sample tested. I'm sure all of us have gone through it, wherein we had to wait for a couple of days to even get the sample tested. And this scenario was not helped further by the availability of only imported COVID kits, which were not enough to suffice the high number of samples being tested in the country. Fortunately, at that time, the diagnostic companies that were mainly into manufacturing of routine diagnostics like biochemistry, hematology, immunology, went into an overdrive, and we saw the emergence of even little-known diagnostic companies coming to the forefront in providing ICMR-approved kits to the Indian market. This challenging scenario pushed all of us to emerge as indigenous manufacturers of molecular kits, especially for COVID. Hopefully now we are entering the phase of eradication of outbreak, and I'm keeping my fingers crossed on that. but it now opens up new avenues of molecular testings. In the past, when a patient was admitted for fever, a symptomatic patient, the patient would be tested for malaria, dengue, chikungunya, typhoid. Now it seems that COVID testing will become a part of this panel. So companies have already started introducing such multiplex panels that will become a part of the routine testings whenever a patient is admitted with a particular symptom of fever and cough and cold. During this time, we observed that nearly 3,000 labs, the exact number is 2964 labs, were given approval by ICMR for COVID testings. Laboratories immediately went into an overdrive and set up the infrastructure for molecular testings. The need of the hour has made every other lab an ICMR certified RT-PCR COVID testing center, and other manufacturers a CDSCO certified RT-PCR COVID kit or OEM indigenously manufactured. But do all these 2964 labs and sizable number of manufacturers meet quality control and quality assurance? That is the question that has to be really asked. To be compliant with the current regulatory framework prior to actual patient testing, all molecular testings and tests need to be extensively verified due to the multitude of options available to the lab today. LDT, laboratory defined tests, ASR, analyte specific reagents, REO, research use only, IVD, CE-IVD, FDA approved. These are the certifications that have to be looked into. We saw in the beginning of the pandemic, a REO product like RNA extraction, which is an integral part of the COVID testing, came under the purview of CDSCO just on the basis of the importance of its usage in a patient setting for COVID testing, where the step of RNA extraction was critical to a correct report being handed out to the patient. A very strong QC, quality control, quality assurance program, both internal and external, is necessary to detect issues, reduce errors, and eliminate problems. Quality management principles relate patient results to prior results or absolute values comparable across time and methods by using international standards and reference materials. Till now, molecular testing has been a forte of chain labs and big hospitals. It is to be seen whether these newly opened nearly 3,000 labs are able to continue and sustain the molecular testings for want of samples for other parameters once COVID testings are reduced. The silver lining in the current scenario is the advent of Indian manufacturers in PCR testing kits. This has resulted in reduction in prices, and now it seems that molecular testings will become a part of necessary tests, which have been the access of only a few privileged people in the terms of cost and availability. The new laboratories that were introduced during the pandemic has ensured that even non-metros and rural locations now have access to such specialized tests. And it won't be far when we see a boom in the molecular testing market size as well. Now companies have gone to even the extent of thinking out of the box. I know we are doing it because I'm leading that project for our company also, and trying to introduce both qualitative and quantitative PCR tests for tests which were currently being done on rapid cards, ELISA, or chemiluminescence. So the future looks bright for India patients in terms of accessibility and especially the commercials involved in getting molecular testings done. So that's my point of view on the subject.
Thank you so much, Mr. Bharat.
It's a difficult name to pronounce.
Yeah, it's very interesting aspects you brought, that there are so many companies who are now coming up with PCR-based diagnostics, and definitely the challenge will be how they maintain their QC as per the international standard. And we hope that our Indian biotech industry will be supported adequately by concerned agencies so that it will be taken care of. Now I invite Dr. Srinivasan Vedantam he is the lead of disease program at MedGenome Labs Private Limited. Dr. Srinivasan Vedantam has completed his MS in Medical Laboratory Technology from BITS Pilani and PhD from University of Madras. He has gained close to 5 years of postdoctoral training from Columbia University and New York University. He has been performing both clinical and population-based biochemical studies and basic mechanistic studies on insulin resistance, diabetes, and its complications. Dr. Srinivasan moved to Madras Diabetes Research Foundation in the year of 2012 as a scientist. He was a senior assistant professor, Department of Biotechnology, Shastra University, India between 2013 to 2016. Since 2016, he is working at MedGenome Labs Private Limited, India, starting as a disease program lead to currently its associate director for operations and scientific affairs. He was awarded the Gerald Weizmann Award for Excellence in Research by New York University for this study. He has several publications and patent to his credit. And Dr. Srinivasan, so we can see from his profile, Dr. Srinivasan has donned multiple hats during his career. He was a teacher sometimes back, but then again moved to industry. And he knows both the sides of the coin, how the teaching and basic research works, and now he's heading the industry. So he has a vast experience. And I remember he has visited KIIT a couple of years back and in a meeting, and we had a meeting with Dr. Srinivasan. Now I invite Dr. Srinivasan to share his views on this topic.
Thank you, Dr. Rohan. Rahul, it's a pleasure. I remember, I think it is twice I've visited your campus. And once, I think Dr. Srinivas was the organizer. And it's always a pleasure to visit the campus. And I thank the organizers again to invite me for this forum, which is much more relevant at this present situation. Can I share my slides? I have a few slides.
Yes, sure.
Yeah. Are you able to see the slides?
Yeah, your slides are visible. You can make it full screen. Yeah. Presentation.
So as I also thank my previous speakers to give a viewpoint on the current scenario of molecular diagnostics. And I feel, we are at a stage where there is a rapid rise in technological advances. And at the same time, there is a market need to implement these technological advances where we can utilize for diagnostic testing. And more importantly, the purpose of doing all these tests is that, how efficiently are we able to implement these technologies, how efficient it is for in terms of the patient outcome, whether these tests are giving us the best yield when it comes to a doctor's perspective and coming out with a positive outcome in clinical practice. So there are two important questions, I think, which was highlighted to be part of this forum that we felt that we need to discuss in detail. One is this, how difficult and technically demanding is the quality control and quality assurance in case of molecular diagnostics. I think quality is it goes hand in hand, right? Unless and until we are pretty sure about our quality and we have important checks, confident about what we offer, the brand doesn't take us anywhere. I think if we will leave out the diagnostic field, but let us go to any other segment, whether you talk about Apple or Samsung or whatever it is, we talk about the quality there, right? So quality can never be compromised. That is one thing. So when we talk about quality aspects, when it comes to molecular diagnostics, it is imperative. It is extremely important that we pay utmost at most we are conscious about what we do about this, and then have separate departments that put efforts in maintaining the quality of the test that we offer. Now, when we talk about quality, quality is always like when everything goes fine, it is always invisible, that we may not even know that we are compromising on certain aspects of the quality. But at the same time, when it goes bad, we cannot ignore it, right? We have to really understand the gaps, understand what exactly went wrong, and then look back, go to the basics to put some matrices which we can follow, so that the department follows the quality aspects, right? When we talk about molecular diagnostic testing and the quality parameters, and is it really now these are all the different parameters that we need to first understand. And now when we talk about molecular diagnostics, it can vary anywhere from a simple PCR based test to the next generation sequencing test that we offer, right? Now, the quality parameters that we set should be uniform, whether it is a small test or it is a high-end test. The quality parameters should be uniform. The quality indicators is uniform. The team is aware of those quality indicators, right? And we have been we should set these quality parameters that are scalable. So if you look at MedGenome 6 years back, we were looking at testing close to around 100 samples a day, but today we are doing close to 3,000 samples a day, which includes the COVID testing too. And it ranges from a simple RT-PCR testing that we offer for COVID to the whole exome and the genome testing that we offer through the NGS-based parameters. So how do we set these quality parameters? And at the same time, we don't burn our fingers in terms of the cost aspect, right? Now, when we look at these aspects, these are some critical parameters I have listed out. One, yes, lab design as per molecular lab requirements. This is again, it is under our control. There are set protocols, set requirements as per CAP or NABL, where they ask for these requirements, and which can always be invested on. The infrastructure can be set up. But one of the greatest challenges that we always face is the qualified personnel. This is something, across the globe, people are facing these challenges. And other important aspect is when we come to our nation, when we come to India, I feel, this is one area where we need to really put our efforts in training qualified personnel who are industry ready and who are able to, it's not only about the technical aspects of it, but it is also about the other soft skills, their ability to handle stress when it comes to a diagnostic industry, because there it is more dependent on you are already in a service sector, and you are answerable to the patient, you are answerable to the clinician, and more importantly, at the end of the day, you maintain your timeline to deliver the results. And all these things need to be done without compromising on the quality aspect. So this is one thing which is very critical. Then validation of the tests. I think this is one of the major aspects. I think, some of the best companies in the industries have really, have grown because that, they are able to come out with really valuable products, quality products, which are validated. Suppose, we come out with a genetic panel or say a non-invasive prenatal testing. These tests have been validated in our population. And, there are some good scientific papers that has come out of it, where we have defined certain quality parameters, its sensitivity, its performance variables, like specificity, sensitivity, how effective is these tests, what is the yield of these tests. These are very critical aspects. Another important thing is control of pre-analytical variables. This is again something which is very critical, at least, in my experience in the past 5 years, we have seen that pre-analytical variables like proper collection of the sample, proper transport of the samples, transport these samples in appropriate temperatures, getting the consent forms, which are extremely important when it comes to genetic testing. And then, when we get this information in the lab, when we access these samples, we see that, the identifiers doesn't match. So these are very critical things. And why it is extremely important and why it is critical is that when these things are taken care of, I think 50% of our downstream operational efficiencies will improve. Say for a simple example, when we talk about genetic testing, you collect a sample from a patient, and then we make a simple mistake of naming the gender. Instead of a male, we put a female, and then the sample comes to the lab. And then when we do the genetic testing down the line, when we do the bioinformatic pipeline analysis, it flags of showing that there is a gender discrepancy. So the cost involved is very high, right? We are talking about tests where we are doing offering it around 15 to 20,000 rupees. And when you at the end of the day, you come to know that there is a pre-analytical error due to which this discrepancy has come. It really hits hard. Other important thing is control of analytical variables, and control of analytical variables again is within our control. I think having proper QC checks, when you extract the DNA, looking at the DNA, RNA QC parameters, and then you have QCs for library preparations, QCs for data quality, QCs for reporting, the probability at which you can actually come with the right variant analysis, or the identification of the right organism, or in fact, the application goes on and on. So setting up QC parameters for analytical variables is extremely important. And of course, quality control at each of these steps will have a greater impact, when it comes to our efficiency. And how do we achieve all these things is that having a proper proficiency testing, we can have tie-ups with international organizations as per the CAP norms, or from the American Society for Histocompatibility and Immunogenetics for HLA typing. You have European forums where you can actually set up these tie-ups where you can have a program for proficiency testing, periodic quality assessment and systems. So we have a fortnightly meeting with the team where they need to present on the number of samples tested, looking at the pre-analytical. You can dissect into each of these departments, right from the human resource personnel, from the logistics team, the procurement team, the analytic, the sample acquisition team, then the lab team. You can set up some quality indicators, and if they are actually complying to those quality indicators, set proper guidelines. Post-analytical management is extremely important when it comes to quality measures, especially in molecular diagnostics. And when I talk about post-analytical management, this is about once you release the report, how do you manage the patient? Are we offering? Is our report giving the best utility to the clinician and also to the patient? And whether appropriate genetic counseling is available, whether the confidentiality of the report that we have shared is maintained. So these are some of the important aspects, that we need to look into, when it comes to molecular diagnostic testing. So to summarize this, the quality indicators that we talk about, I think there should be proper. I've just summarized this in a simpler way. I think in the pre-analytical part, we always look at proper sample labeling. Are we able to release the reports on proper turnaround time, error-free reporting, reduce sample rejections? Because all these things add costs. And with the current average pricing that we give, except for the RT-PCRs, I think it is extremely hard for any company to take on these costs. Reduce repeat tests and then inexhaustive inventory. I think inventory management is again very critical when it comes to in higher cost tests, especially in the molecular diagnostics industry. So I think I will just stop it here, and I'm not sure if we need to talk about the other aspect of it. I think the pharmacogenetic aspect can be discussed separately, right?
Yes, Dr. Srinivasan, yeah, pharmacogenomics, but it will be nice if you just put a little bit light on how the pharmacogenomics aspects are working, because you are engaged in this direction. So maybe just a couple of lines on how things are working. Yes. That will be great.
No, I have a couple of slides on that.
Sure.
So again, I think the second aspect of the talk was on what will be the cost effectiveness of the pharmacogenomic industry look like in the near future. Well, it's a very interesting question. Maybe the person behind this, I don't know, I think I need to appreciate them. But what is important here is to note that it's not only about pharmacogenomics. I would put it under the umbrella of genomics as such, because cost effectiveness of genomics is something which we have been talking about for quite some time. 6-7 years back, when these exomes or genetic testing was launched, we were looking at doing a testing. Let me talk about the Indian perspective. Let me not go into the global perspective as such. So we were talking about testing at somewhere around 30,000 rupees for a clinical exome, and 40 to 50,000 rupees for a whole exome, and then of course 1,20,000 for a whole genome testing. But now, we are able to give a quality result on clinical exome or a whole exome testing at an average pricing of somewhere around 15,000 to 20,000 rupees, right? I think that's where, the technology has come up a long way. And if I now specifically talk about pharmacogenomics, I think the same holds true. So what is that for the student audience here? What I would like to emphasize here is that, what do you mean by pharmacogenomics, right? So we are talking about, importance of certain genetic variants that might actually impact the metabolism of drugs. Whether, we know that some of these genes are actually enzymes that are involved in the metabolism of these drugs. Like how do these drugs metabolize? They involve in the pharmacokinetic and pharmacodynamic properties of these drugs. So based on these genetic variants, association of these variants to a particular drug can actually classify or categorize the patients into slow metabolizers or normal metabolizers, rapid metabolizers. I will give you, for the interest of audience, especially the student population, let me give you a simple example of tacrolimus. Tacrolimus is an immunosuppressant, right? And they are generally given for patients, especially for organ transplants, in kidney transplant or even during the bone marrow transplant. The recipients are subjected to tacrolimus. And tacrolimus is metabolized by an enzyme which codes, it is a gene which we call as CYP3C5. Okay, and specific variants in these particular gene can actually, help us categorize the patient, whether this patient can metabolize tacrolimus at a faster pace. So depending on that, do we need to adjust the dosing of the tacrolimus drug to these patients? So the same holds true for several other drugs. We have long-lasting implications in the cardiovascular area, where cardiologists, especially give thrombolytics like clopidogrel, warfarin. We have great implications in statin therapy. Certain people metabolize statin in a different way compared to several other people. So the horizon of pharmacogenomic testing is increasing, but when we talk about some of the factors that actually are having an impact when it comes to pharmacogenetic testing, why we still are in a stage where a lot of them are still not utilizing this pharmacogenomic testing, is that one is the clinical utility, right? When I talk about clinical utility, the clinician would like to see a concrete evidence that if I perform this test, I am able to adjust the dosing, and I am able to have a proper therapeutic management, right? But most of the pharmacogenetic testing of late has been more involved on the research aspect, right? We have been talking about several research papers that have shown utility of these tests, but then when it comes to practical challenges, there are lack of clinical trials and there are lack of clinical research. And it is not only that, it is also, ethnicity driven, right? It is not like, Indians behave, their genetic makeup is different. They behave in a different way. The Caucasians' genetic makeup is different. So, their response to certain drugs are very different, right? So this is one important thing, I think, where both the scientific community and also the industry, especially in the genomic space, needs to put efforts on to make the clinician understand that it has a clinical utility. Now, when we talk about technology, the cost, as I said, it holds true when it comes to genomic testing, right? We have been utilizing the NGS-based testing. Well, today, if you want to look at clopidogrel response alone, we can implement a RT-PCR test to identify a response to clopidogrel. But does it have a value? Well, that is more of a reactive testing, right? How do I know that an individual responds this way to a clopidogrel, right? Generally the cardiologists who have the patient with him who has undergone a surgery and he wants to start with a clopidogrel, he might ask you to do a clopidogrel testing. Now, the cost of clopidogrel testing through an RT-PCR is somewhere around 1,500 bucks to 3,000 bucks now. But what is very important is that if we can multiplex, if we can leverage the use of genomic technologies like the panels, the exome panels, or the human genome testing, to completely understand whether these individuals how are they responding to a particular set of drugs, right? That's where I think, the focus should be. And that's where the utility of these tests are. And that's where, we can cut down on the testing cost, right? Now, as I told earlier, clinical research and validations are extremely important. Awareness is something, at least I strongly feel that in countries like India, I think awareness is very important. We have come a long way. It's not like 2015 or 2016, but now 2021, we are able to, penetrate the tier 2, tier 3 cities where the clinicians are aware of many of this genetic testing and the importance of these pharmacogenetic tests. But then, cost is again one of the major factors I think, the industries need to think about. So when I talk about technology here, bringing down the cost, I also say that I think we need to have some, genomic arrays where we can cut down on the cost. I think, one of the important things that we as a company did was through our Genome Asia 100 case study, we were able to develop genomic arrays, where we are now able to cut down the cost on pharmacogenetic testing from anywhere around 15,000 to 20,000 rupees using a panel to using arrays, where we are able to offer it close to around 4,500 rupees. And we do close to around 18 genes, which is relevant to 49 different drugs. So that is on the technology front and awareness front. And if all these things come into picture, and if we still feel that there is a great clinical utility, and the insurance companies and the clinicians feel that it is no more an experimental test rather than a utility test, I feel that the insurance companies will fall in place. If this starts to, when it comes to India, I don't even talk about insurance, because I don't know how long it takes for us to come under the insurance ambit, especially for my genetic testing. But at least in the US, I feel that there are 1 or 2 players who are getting into it, especially from the public-private fund. But I am sure that in the coming years to come, they will have a bigger role to play. So there is a huge impact of genomic testing, especially when it comes to pharmacogenomics. I think the right mix of awareness, right technology, right understanding, and offering products that are relevant to that particular ethnicity is what is the key, where we can cut down on the cost. Thank you, Rahul.
Thank you, Dr. Srinivasan. I'll take a couple of questions. First of all, I'd like to thank Mr. Bharat, because he has already addressed many of the questions in the chat itself. But one question I feel really is interesting, which will be for both of you. So I'd like to divide the question into 2 parts. The question is how the IPR protocols or IPR domain is reacting to this new generation testing. So there are 2 aspects. One is IPR on products like RT-PCR test kits, etc. I request if Mr. Bharat can throw some light on IPR issues in developing certain kits. Mr. Bharat, are you?
Yeah, sorry. I did not realize I was on mute.
Sure.
See, it's in today's diagnostics market, it is very difficult to have a patent. That is the reality. Most of the companies which are developing new products in India are following the traits that have already happened. So there is no patent available. We are just, I can talk about my company. We are just working on the formulations that have already been done. Unfortunately, we still do not have good companies in India which manufacture raw materials. So we are still dependent on procuring the primers or the master mixes from international companies, especially the primers. We do not have them very much commercially available. And most of the kits that we develop are non-patentable because they are the same kits that are available in the market. One of the answers I gave to regarding pharmacogenomics was the drug Keytruda. Keytruda is a drug for small cell cancer, lung cancer. And this is a drug developed by a pharma company called Merck. And the corresponding assay, I'm sure Dr. Srinivasan would know more about this. The corresponding assay of PDL1 is developed by a company called Dacoagulent. And these go hand in glove as far as the treatment of lung cancer is concerned. And the PDL1 has a patent. So it's a very novel kind of a product with a particular target size, target sequence. So they have a patent. So anything that can be developed to that level, which looks difficult at this moment to have indigenously manufactured in India. So I think most of the time, see, you have to understand the reality that India has never been a molecular testing hub till now because of commercials, because of various reasons and availability and various other factors. So right now we are following a trend which has already happened. So IPR in indigenously manufactured kits really does not play a very important role at this moment.
Okay, thank you. That's a very interesting domain, because even India is a player on the generic pharma market. So looks like even in the diagnostics also, you are going to be, so far we are going to be a generic player, right?
Yeah.
Okay.
No,
yeah.
Dr. Rahul, I would like to just
Sure,
add a point.
sure.
So as Mr. Bharat said, see, I would like to classify this pharmacogenomic, range of tests or the products into two different types. One is your companion diagnostics. Okay.
Yeah.
The other one is your personalized medicine, where you can always go to anyone, anybody of interest. They can always give their blood samples to see, if they have any variants that are associated with any drug. Right now, the value for any IPR is in the first thing, the companion diagnostics.
Right.
Now, is India there in that portfolio? No, but I feel that several companies who are now in the place, like you have really good companies, who have invested more on the R&D aspects of it, and I'm sure that there is always a scope. It is just that the mindset that we need to train. I think we have the best of facilities. We have, we are, come on, we have been talking about rare diseases, and then, I can show you how many, though they are all not rare, but, it is because of our capabilities that we are now able to fix a diagnosis for all these cases, right? Whether the drug is available or not is a different thing, but yes, so the infrastructure, the intellectual capabilities, and the will is there. It is only that it is the mindset, I think, that we need to move into more of a product oriented approach to a service oriented approach is what is lacking.
Agreed.
Yeah, agreed. That's a very interesting point. So, Dr. Srinivasan, my question to you in this regard is, if you develop a process for, say, genomics applications, can that be IPR protected? Like, say, for example, if you develop a cancer diagnostic panel for a particular cancer, which is kind of India specific, say, for example, oral cancer, can it be IPR protected?
It can be, but the challenges are the consent, the confidentiality. See, one of the major challenges that we face, especially with the genetic testing, is that we don't have a proper policy on the data security.
Right.
We have proper policies on what is how are we, like, there is no uniform way where this data is collected. What is the utility for the patient? What is that, especially the ethical aspects, that needs to be considered when it comes to genetic data, and how uniform are these data, right? I think, the focus must be more on these areas, setting up these processes. And of course, yes, if all these things are addressed, I feel that yes, there is a value there for an IPR.
Okay, thank you. So last question for this session, I will ask. So how do you, to both of you, like you can share your views, that how do you train or prepare the students to come into the diagnostics domain in their future? Like, there are different aspects. So how basically a student, an undergraduate student, should prepare himself or herself to enter in the diagnostics domain? So what are the basic qualities or skills they should try to acquire?
Right.
Okay.
Yes,
Mr.
Srinivasan, you would like to go first?
Yes. So one of the important aspects, what I would seek from anyone, especially from the student community, is that stick on to your fundamentals, right? When we hire people here at our company, the first thing we ask is, how do you do a DNA extraction? People think at the bigger, NGS, they think about, sequencers and all those things. But what they don't actually focus on is, the fundamentals, right? At the end of the day, we'll again look at fundamentals. One. Another important thing is what I feel the student community needs to understand is the soft skill aspect, right? When I talk about the soft skill, it is that what is their endurance for stress? In an industry, what is important is that, how you handle the stressful situations. There are, in everyday, day-to-day aspect of any service industry, the first thing and that too, when it comes to some of these critical testings, well, genetic testing takes around 2 to 3 weeks, but when it comes to a short range test, when we talk about COVID PCR testing, or when we talk about a flow cytometry testing for some of the leukemias, hematology range of offerings, I think these are all dependent on the short pack, right? The sample comes in within few hours, you need to, come with the answer. And if something goes wrong, they start to panic, right? So these are certain aspects I think, the students need to be trained. These are all mostly on the soft skill aspects. And another important thing, what I strongly feel, is the attitude, the willingness to learn, right? You may not be extremely brilliant, but then, that single aspect of, willingness to learn, and then, be a team player makes, a big difference when it comes to, industry perspective.
Thank you. Mr. Bharat?
Yeah, this is a very nice topic, which I always talk to my, I'm a part of the alumni in my college as well till now. And I fully agree with Dr. Srinivasan on the fact that fundamentals are important. The clarity, my still go-to book is the Stanier, and I still refer to it for all kinds of genetic, and I continue to refresh myself even after 2 decades from after started to work. It is very important to understand, I'm talking about the undergraduates, where you want to see yourself. A self introspection is important to understand whether you want to continue with research, or you want to go into product management, or you want to go into sales. And that is an introspection a kid has to understand before they take on a career. Because if you're not satisfied with what you're doing, you're not going to enjoy. But having said that, for all kinds of the career later on, be it in research, be it in a laboratory, be it in sales and marketing, your fundamentals are very, very important. You have to go on learning on a daily basis. You come up with different technologies, you meet people from different aspects of the industry, and your fundamentals are very important. So I always say that you have to make up your mind where you want to see yourself. I took a conscious decision of not going ahead with my research after I passed out from ICMR, because I did not think I was mentally, I was going to be satisfied with doing research in my life. But then there are people who would like to do their, and if you want to go into research, I would always suggest, do not stop before a PhD. A PhD is a very important aspect if you're going into research. I'm sure Dr. Srinivasan will agree with me, because it actually increases your strength for lab work. Most of the students who pass out have done their masters by paper, and lab work is a little bit part of it. Whereas a PhD, when you're actually writing a thesis and doing the experiments, it's very, very important. So it all comes down to the mindset of the kid, where do they want to see themselves, and then choose a path accordingly. But if you are coming into sales and marketing, I would always suggest complete your master's. Do not stop at graduation. Master's is a very important degree, even when you come into sales and marketing in this field also.
Thank you so much. Thank you, Mr. Bharat, Dr. Srinivasan, and Mr. Dubey for your excellent deliberations and addressing all the questions that is there in students' mind. I will now, its time is almost over. I think we already overshot the time by a couple of minutes, but that's all right because we had a lot of great discussion. Thank you once again for accommodating our request and giving us time and giving our students to listen to you, although in a virtual mode. And we hope to see you again on our campus in the actual physical mode very soon. Thank you so much.
Thank you, Dr. Rahul. Thank you so much for having us.
Thank you, Dr. Rahul and Dr. Gopal.
Thank you. Now I invite Dr. Amrita Mishra, Assistant Professor at KIIT School of Biotechnology, to continue with the next session. Just a few words about Dr. Mishra. She has more than 12 years of experience in biogenic nanostructures processing for applications in wound management and infection control. She completed her PhD from Cheongbuk National University, South Korea. Her research work is also on isolation and characterization of biomolecules for potential antibacterial applications against MRSA. She is a member of Association of Microbiologists of India. Over to Amrita now. Thank you.
Thank you, Dr. Rahul, for that introduction. And I hope I am audible to everybody.
Oh, yes.
Okay. So we begin with the session 2 for the day 1 of the Industrial Conclave. So good afternoon to all the dignitaries, invited speakers, faculties from School of Biotechnology, and all the dear students. The theme for the second session is Data Analytics Tools Turning Information into Insight. The aim of collaborative data analytics is primarily to integrate people, information, and processes. Hence, encouraging collaborations across departments with R&D, quality, statistics, and validation is important in today's pandemic period. It will also help support a future in which ways of working will be probably different. Hence, organizations have gravitated towards predictive analytics in the last several years as they use data to anticipate future trends and needs. With this being said, I, Dr. Amrita Mishra, Assistant Professor at School of Biotech, KIIT deemed to be University, welcome you all to the second session of the Industrial Conclave. I would like to introduce our first speaker of the session, Mr. Ranjit Prayan Kotvitil, who is working as Senior Director Biostatistics and Statistical Programming, Pharmaceutical Products Development, India Private Limited. He is heading the department and overseeing India operations. Mr. Ranjit has more than 15 years of research experience and has previously worked in industries like Novartis and Ventive. He has vast working experience in therapeutics and clinical industry and has a strong background in statistics and programming. I request you, sir, to kindly address to the audience and begin the session. Mr. Ranjit, if it's okay, can we start the session with you?
Sure. Thank you, Amrita. Hope all of you can hear me properly.
Yes, sir, we can.
And thanks for the warm welcome. And I would like to switch off my video because maybe that will impact the bandwidth and create some troubles. Okay. So as she mentioned, our topic is about the data analytics, and I think all of you will be hearing a lot about data analytics nowadays, because if you look into somewhere or if you search into the LinkedIn or opening for data analytics, and the world is moving towards that, because the accessibility of data or the availability of data is increasing day by day, right? I think all the speakers here, we all are from the clinical background or from the pharmaceutical industry. So most of us will be talking or providing the examples or inputs based on our industry. We used to conduct the clinical trials within with lots of restrictions, where we do select the samples from the population, then we check the inclusion exclusion criteria. So like that, our recruitment process happens, and the success rate of getting a product into the market was very, very minimal, right, due to all this restriction and everything. And now how this data is helping us, because of this data flow, we started doing the real world analysis studies, and that is like helping us to get a more products into the market, or that is helping us to increase the number of samples. I can very openly tell you, by 2022 end, as per the information, the 1.7 megabytes of new information will be created every second for every human on the planet. That means your data and my data, we are knowing or unknowingly, our data are like passing, right? Most of us will be wearing here Fitbit, right? When we are wearing Fitbits and going for a walk, what is happening? Our maybe height, weight, BMI, respiratory information, cardiovascular information, this all data will be sharing with a big organization. Maybe that is with Google or maybe with Oracles, okay? Like that, data is flowing, and the availability of data and the accessibility of data is very, very huge nowadays. Even if you Google it, or as per Google, they say in our world we have more smartphones than a toothbrush. This is the current data, right? It's very interesting, right? Very interesting to listen. We have more smartphones than a toothbrush. So we are moving towards that world. So data flow is increased, data accessibility is increased. Now, how can we utilize this data in a more better way, and that will help us to get the drug into the market as early as possible? So that's what we are doing. That's what we are like, depending the data analytics or data analysis. The one of the issues that we face in clinical industry is cost and time, right? Cost is increasing day by day for the drug development process, and it's taking too much of time as well. We have the current example or experience in front of us, pandemic, right? We all were waiting for vaccine. We all were desperately waiting for the vaccine. And the people who are not in clinical industry, they used to ask us, why vaccine is not coming? What you guys are doing? Because they doesn't know what all are the process that we need to follow. We are playing with the patient's life. We're dealing with the patient's data. So we can't just get a product into the market. We need to prove the safety of the medicine. We need to prove the efficiency of the medicine, right? For that, this data helps us a lot. And I told about the success rate. Maybe like in a year, if we are like all the pharma organization together, if we are submitting maybe 100 applications to an authority, 10 comes out to the market. So success rate is very, very minimal in clinical industry, right? Just think that 10 out of 1,000 drugs only coming to the market. So very, very low success rate. And there are different reasons for that. And sample is one of the reasons. The samples, we need enough patients, right, to conduct the clinical trial. And we struggles a lot to get the enough sample size, the participation. So now when we have all these gadgets, when we have all these sensors and all those, that is helping us to get more data. We don't need to depend. We don't need to tell patients to come to the clinic, right? Patients, because some of the patients, or the volunteers, human volunteers, or the healthy volunteers, they were hesitant to participate in the clinical trial because they have to come to the clinic, they have to provide their information, okay, travel. There are lots of challenges like that. But now, due to these gadgets, due to these sensors and all, sitting at the home, they were able to provide their data. So data, that's what I'm telling you, data flow started increasing. Recently, the one of the US-based organization, Eli Lilly, they came up with a digital inhaler. Okay, what patient need to do is just put that inhaler in your pocket. and all your respiratory parameters after using that inhaler, all your respiratory information, this using the sensor that inhaler will capture it and pass it to the organization, and they can analyze the data for a respiratory studies like for asthma, for COPD, like that. So data started flowing, and as I mentioned earlier, another reason for the failure was when we were conducting the clinical trial, we are selecting a sample from this population, and this sample is having lots of limitation because we do check the inclusion criteria, exclusion criteria. So that means we are scrutinizing, we are categorizing the data, we are like checking everything, and then selecting a small sample from this population. So maybe that drug will work or the drug will not work for that particular samples. Now what is happening, due to the sensors, due to this targets and all, data started flowing to us, and we started using real world data. There is no inclusion criteria, there is no exclusion criteria. The people who are like daily using this type of sensors target, we started getting data from them, and then we are able to conduct the clinical trials into the real world, and then the success rate started increasing, or we are getting enough data to do our analysis. And these data are now helping us to come up with the personalized medicine or targeted medicine, right? In future, you will see now what is happening. There are lots of drugs into the market, right? And anyone can go and have it. But in future, it is going to be personalized. The medicine maybe that will work for me, that will not work for you. And utilizing such data, the organizations will be working towards the personalized medicine and targeted medicine in future. And this data analytics is helping us to come up with lots of tools like a patient data dashboards, the graph that will help us to understand the safety signals in early stage. Now we have remote site monitoring. We don't the CRAs, they don't need to go to each and every clinic and provide the information, provide the data. Sitting at home or sitting at office, they can monitor the site and capture the data. So like that, these data flows are more, data accessibilities are more, and that is definitely helping us a lot to get a product into the market as early as possible. That is helping us to reduce the cost and reduce the time. Okay, so as an introduction, or maybe at the beginning, I can briefly talk about this in this way, and I know my friends are there, Abhishek is there, Shekhar is there, they will talk further about it, and then definitely the floor will be open for further question and we can further discuss about it.
Thank you, sir, for that interesting introduction. You have, as rightly you have told, that with the increase in data flow, the product is reaching more quickly and the commercialization is becoming more easy, whereas there were a lot of restrictions before when and that is the reason probably why the significance of data analytics is gaining attention across the domains in different industries. So thank you, sir. I will now introduce our second speaker of the session, Mr. Abhishek Mishra, Head of Programming Development, GSK India Global Services Private Limited. Mr. Mishra started his career as a statistician in Pfizer Pharmaceuticals India Private Limited, where he was part of global modeling and simulations team and worked on various simulation projects. Prior to joining GSK, Mr. Mishra has worked in Novo Nordisk Service Center Private Limited for more than 7 years. He has established and led many teams of statisticians and programmers. He has more than 15 years of experience in clinical domain, including 9 years in people, project management of leading teams in global work environment. He has experience of working across different therapeutic areas. I request you, sir, to share your views and thoughts with us.
Am I audible?
Yes, sir.
Okay, thank you, Mr. Gopal and Ms. Amrita, for inviting me to this particular event. It's really an honor. And I'm going to talk briefly about data analytics tools turning information into insight. And I'm going to focus on primarily two aspects in my talk today. So, like Ranjit mentioned about different sources of data and different advancement in technology. So I'm also going to talk about these two aspects. And the first aspect is advancement in technology and also availability of these open source softwares. And you all would have witnessed that there have been a lot of advancement being made in technology in recent time, like Ranjit talked about digital inhaler. Similarly, now we have watches from which you can take calls, you can count your number of steps, your sleep patterns, your vitals. All these things are kind of can be captured and collected like by wearing a simple device on your arm, and then from the convenience of your home. And I always tell my team that one of the positive side of this pandemic, of course, there are so many negative sides, but if I have to talk about one positive side, then I would say that this pandemic has actually further pushed this determination within industry, governments, and other organizations to further innovate and adopt cutting edge technology. And that's actually have gone up. And same thing is also true even for pharma and biotech industry. Now use of all these fancy terms we used to call like artificial intelligence, machine learning, natural language processing.
Dr. Rahul, I request you to kindly mute yourself. Dr. Rahul? Dr. Rahul, could you mute yourself? Yes, okay. Sorry, sir, for the interruption.
It's okay, sorry. Yeah. So data mining, robotics, these things are nowadays reality in pharma and biotech industry. And these things are being used both at research level within labs and also in development of the drug. And this investment has also bring more efficiencies in operations of the pharma organization, as well as also a kind of reducing the possibility of human-induced errors. And we also would have heard a lot about this predictive analytics. And it's not like this that this is a new term. It's been in practice since quite many years, but with new technologies, with high processing computers and with other softwares, this has become, I would say, this has been practiced more into reality. And this is nothing, basically, this is more like how we can use the historical data to predict the likelihood of outcome in the future. And like Ranjit mentioned, that clinical trial is a very expensive and also time-consuming process. And these things actually help us a lot to determine which molecule will move to a next phase, which molecule is safe, which molecule is efficacious before doing the actual trial. And when Ms. Amrita spoken about modeling and simulation, so when I used to do simulation, basically one of my job was to kind of to predict some scenario based on the simulated data, so that you're more confident when you are going to do your clinical trials. So this predictive analytics is certainly kind of gone up. The other thing which I want to talk about is the open source software, and you might have heard a lot about this terminology and also about R. In fact, if I can quote my own example, that when I was doing my MSc in statistics from Pune University, I used to have one particular subject called statistical analysis in human genetics. And I'm sure you guys must be knowing that this gene expression data is very huge, and it's very difficult to handle this data by the normal softwares. Even the Excel files cannot store all the data and so on. And the open source software, and particularly R, actually kind of helped, at least to me at that time when I was doing those projects. And now you can see that these open source softwares, R, Python, are being used very widely. And the beauty is that there are so many packages available. You can also create your own package if you want to do a particular analysis. And you can create graphs, you can do statistical modeling. So all this is kind of becoming very simple and accessible because of the open source software. The other aspect that I want to focus today is on the data visualization tools. And in a lot of pharma and biotech companies, there is a focus on having more of data visualization tools within the organization. And what actually it helps is that typically you need a programmer or a statistician to do any kind of analysis or generate some graphs. But by having these data visualization tools in place, even a clinician, even someone who doesn't know ABCD of the programming can produce graphs, can look into the outliers, can see different kind of trends into the data. So it also helps into the data democratization, wherein the statisticians and the programmers are not the only custodian of the data. It's kind of available for a wider audience, and it also brings in a lot of efficiency, because otherwise the programmers needs to produce those outputs, and then there's a particular process someone has to develop, someone has to review. So by having these tools, they can just kind of produce just in case kind of analysis and look into the data. So I would end my talk by concluding that due to recent changes in technologies, a lot of tools are available freely and can be used by anyone. Thus, you don't need to be a programmer or a statistician to get insight out of the data. And I would encourage all of the students to learn how to get insight from the data that you deal with, to connect all the dots, and also to kind of conclude on the basis of data and not perception. So that's it from me. Thank you.
Thank you, sir, for sharing your views and ideas with us. It was very informative. And as you rightly told, simulation, predictive analysis, these play a very important role, particularly when we are doing experimentation. And many of the students are actually curious, and they have asked a lot of questions in the chat box also, because they would want to know, like, where do they stand if they want to have a career, if they want to go ahead with this, and how much they need to learn if, from an industry perspective. But however, we'll be taking up all the questions at the end. So I would now move on with our next speaker for the session. I invite Mr. Shekhar Ayagari, CSPO, Product Owner, Business Process Expert in Novartis. Mr. Shekhar completed his undergraduate and master's in genetics from Osmania University and his postgraduate diploma in bioinformatics from Savitribai Phule Pune University. Mr. Shekhar has over 13 years of experience in delivering and implementing conventional and SaaS-based life sciences, biotech, pharma, and healthcare solutions. Thank you, sir, for joining the session with us. I request you to begin with the talk and address us.
Thank you very much. Thank you for the opportunity. And, it's really an honor to speak with colleagues and co-professionals in the same domain. Right? So after hearing Ranjit and Abhishek, right, I would want to probably talk about a slight different aspect of the same thing. It's like the other side of the coin, right? We talked about what is possible, but with the technology that is available in terms of what is possible, now I want to just highlight what is a potential rate limiting step here, or what is our hurdle, right? Because that also is something that we really need to recognize. And most of the cases, it is known to us. We have been doing it, but we just don't correct this in most of the cases, right? So if it's okay, maybe I will go ahead, share my screen. There's a small presentation that I have just put together, maybe just to help me also go through it, right? So let me just bring it up. And of course, by virtue of my association with Novartis, I have made every effort to basically use the information that is from my experience, what I've seen. I have been a life sciences guy by education, but from an experience, I've been a business analyst who helped in putting IT solutions into various laboratories and healthcare pharma firms across the world. And as a part of that, right, what I have always seen is challenges associated with data, certain processes that we follow, which inherently push us into the corner, not giving us much wiggle room, right? One such thing is life sciences side. We sometimes use a lot of different pieces of software. In some cases, we rely more on the lab notebooks in our laboratory to capture our results than a LIMS kind of a software. As a result, what happens is some of our data is stuck in like images, some of our data is stuck in Excel sheets, some is there in a lab notebook. The first challenge is going to be to find the lab notebook, then find the page where this data is before I could get to it and do anything meaningful with it, right? So when we take this whole thing and then we'll just look at an overview. So here this slide would be more like, definitely not an attempt to teach a fish how to swim, but just highlight what we are looking at. I mean, as a process, when we look at it, right, if we look at this equation, this is the equation every single healthcare or pharma professional or pharma company is fighting. You get 20 years. That's the life of your patent from the time you discover something. If it takes about 13, 14, 15 years to develop it, you've got only 5 to 7 years to retrieve on the investment. But what are the other problems that complicate it? The costs are very high. Research doesn't come cheap, right? The risk of failure is also high. Like Ranjit said, Abhishek highlighted, the number of drugs that actually get in and what comes out of the process, right? That itself speaks volumes about the rate of success, which kind of corresponds to also the higher failure rate. Takes a very long time. Return of investment is questionable because even if I come out with a product, right, there are going to be pricing pressures. I can't really price it in a way that helped me retrieve all the amount that is sunk into the research and development of this product as well as the other failed compounds along with it, right? Unless and until that is retrieved, there is no possible way you can actually reinvest this to go for newer products, right? So these are problems. Now, what is another alternative if I don't want to basically start from the scratch and do a de novo kind of a discovery process? The other thing is like I could do a drug repurposing, right? What does it give me? It can give me a quicker outcome. The success rates could be low, could be questionable sometimes, but at the same time, it's not very expensive, thus making it an approach or something that we can take a stab at it to see what we get out of it, right? And in the COVID pandemic season, we've really seen anything and everything that we felt could be relevant to deal with these kind of RNA viruses have been used in the treatment regimen, right? Not a very scientific way of drug repurposing, but at least a certain attempt was made. Return of investment, again, the costs are going to be low, so it may not be such a dicey situation with regards to what we get it, right? And the bigger benefit is like it will really help me save on time because there is a lot of information that I could potentially reuse existing information. Now, when I take an existing drug for which I have gone through a clinical trial, through the entire preclinical, looking at it from a different indication, this data does not change, right? The question I posed or what I want to use this drug will not really affect how the drug performed and fared in my preclinical trials. That data is still valid, right? How the drug did in certain parts of the initial clinical trial, again, some part of that information I can still utilize, right? And the rest of it, I can start doing a bridge trial and get to filing faster, right? And something that was either not, let's say we anticipated a particular drug to be a blockbuster. We went after it, but it didn't really shine that well in the market. But if you find an alternate use, now there are two potential avenues on which the drug could work and then can generate revenue for it, right? End of the day, every single pharma company, nobody is really here for charity. Everybody is running this business. It's not for profit. So what revenues are generated really defines like how much gets reinvested for unmet medical needs, right? Now, with these two things having talked about as certain options, what can I do, right? Very interesting and easier answer, right? Leverage the tools to analyze data and get actionable insights. I mean, we keep using this like a catchphrase almost in everyday conversation, every single meeting. But the hard reality is that in most of the cases, this is easier said than done. The reason being, in my opinion, every single pharma company out there in this world is sitting on a gold mine of information. Everybody has data, right? And when we say technology and tools are also available, why is that we are not seeing really miracles happening, things turning around at a much faster rate? And that's why I say this is easier said than done. Why? Everybody is having the same asset and liability, which is our data, right? Our data is very distributed. It is collected in multiple stores, stored in multiple places. Like I said, some notebooks, some files, some proprietary softwares, right? Then the whole infrastructure or the whole landscape is fragmented, right? In computational biology, we used to see this case that for my particular activity, I will do my research work and then see what is the best suited open source tool out there, and then I will work with it, right? But that has certain proprietary need in terms of, I need the file format to be this way. You write your own program, you manage it, and then for that point in time, I'm done, right? It's only a little later the whole concept of building a pipeline came into picture, like stitching together these various tools with certain backend scripts running that will convert this format from 1 to 2 to 3 to get this to work in a seamless fashion. For my eye, it looks like a seamless pipeline, but in the background, the formatting is really happening, right? Now, second major question when I talk about assets and liability is really data quality. How clean, reliable, and reproducible is my data, right? Because every time we say that my data is stored in disparate systems and I want to now aggregate into a simple single source, I would have to do some sort of a data massaging, some sort of a data transformation. Now, all of these processes, unless and until they are backed by real science, right? We do not, or there is a risk that we may actually lose the meaning or the insight in that data. It is not an IT data transformation. It is scientific data being transformed, right, into a new structure using IT tools and techniques. And in this process, this is where the core scientists or the life sciences people come in to check and ensure that the meaning is not lost. Otherwise, I mine this data, it will tell me something totally new, right? Then the other item that I have indicated, like interoperability, right? Abhishek indicated there is some data. Let's say if we take about the same example of Abhishek's, right? Gene expression data. Excel cannot handle it. There's a certain limit to it. But the number of genes on a particular array that I have run in one chip, and it gives me that volume of data. And since I can't put it in Excel to do further analysis and I left it in those proprietary formats, how will I even extract that information? What will I do with it, right? I need to do aggregation of data to take all of this data, bring it into standardized formats so that I can pose my questions and then get my insights out of it, right? Now, once I have addressed and turned my data, which tends to be sometimes a liability if I don't follow these, and then make it my asset, what can I really do, right? What's next? I can use this data to drive process optimization, right? I really need to look at my data and understand where exactly I've been slowed down, right? And for me, like, you know, I've looked at multiple aspects of the entire the drug development process or the working of laboratories, and being a business analyst, that kind of put me in this unique position to say interact with lab users, the end users to the super users, understand the various actions that they do, how they do it. And this kind of gave me an insight to say that there's tremendous amount of options or tremendous possibility to drive process optimizations. It's just that use of data should be done to identify the bottlenecks, and then we go towards it, right? Now, another thing, what we can do in the pharma world, I'm running my clinical trials today, but if I am following the exact same rigor of monitoring a site, a good site versus not so good site, like, you know, least risky site, safe site versus not so safe site, why do I get anything out of reviewing a site data coming from a site which has been known to give me good quality data 1,000 times? It does not change anything, right? Rather, I use my limited resources and deploy them where these checks are required. right? Efficient usage, risk-based efficient usage of my resources. And those kind of insights are what are to be drawn using our data, which will help our process become more streamlined, right? What is clean doesn't need to be cleaned any further, right? Save time there, deploy elsewhere, work through it. Then same case with data reviews as well, right? Most importantly, also learn and automate tasks, right? We, from a life sciences background, when we get into a lab setting, we get certain used to some things, right? Tend to have a bit of a hesitation to embrace changes at a much faster rate as the world is changing, as the technology is changing. We're not all there, right? But ideally, we should head there if we really want to stay in the race and win this race, right? And with this, right, I would want to quickly just talk about one example and then conclude my talk to say, now, once all of these things are done, what can be done? Now we can do AI, ML, everything, right? If I have a clean data set, good training set, then I can teach my systems to learn. I can't teach anything, right? And you could see that these are potential impacts, not to mention about the companies, but really where the companies have been leveraging, like drug discovery and research. There's so many of them. Data management, biomarker discovery, diagnostics, pharmacovigilance, patient care, right? Certain examples of patient care have been shared by Ranjit and Abhishek, how we can deal with all of this information, drug repurposing, clinical trials, right? The potential is there. The possibilities are endless. It's just that we need to have the foundation strong, have our data, and have our data scientists ask the right scientific questions. And that drives us in a direction that will help us leverage the advancements in technology and deliver better medicines, better outcomes for our patients. With this, I would like to conclude and thank you very much for your time and patience.
Thank you, sir, for the presentation and also for briefing us about the importance of data analytics and its significance in the industry scenario. Also, you highlighted what are the challenges an industry can face and probable risks involved. And most importantly, cost economics and revenue generation is of utmost importance for companies. So organizations are generating large amount of data nowadays in real time, and subsequently they're looking for professionals also who can understand, visualize, and assess these. So since we have heard from all the speakers, the session is now open for discussion. So I would first like to ask a few questions to the speaker. My first question would be to Mr. Ranjit, that with the rapid need and development of digital ecosystems, how is the biotech and pharma industry coping and undergoing digital and analytical transformation? So if you can share your thoughts on this.
The transformation is happening, no doubt about it, but there are some challenges because it's not like other industry, as others mentioned. It's not like banking, or it's not like insurance or market research where we play with the maybe personal loan or credit card and all those things. Here we are playing with the patient's life. Okay, so safety is the utmost like a care that we need to take care, and we can't compromise quality. So it will happen very slowly here, very slowly in clinical industry. It will take time, but the transformation is happening. That's what I mentioned. I know we have Abhishek from GSK. I know the GSK is doing lots of real world analysis studies now. So that itself is a transformation, because previously, earlier, as I mentioned, in the clinical trial, we were not doing real world analysis. We used to collect the samples or select the samples from the population and check their inclusion criteria, exclusion criteria, and make sure this is the correct sample for doing the trial, and then conduct the clinical trial. But now we are coming out of it and we are ready to experiment into the whole world, that is the real world. So that is one of the transformation. I told about the digital sensor that one organization came up with, where like we can keep it in our pocket and that will provide data. I told diary data, like previously we used to give diaries to the patients to capture the data, like they need to report, they need to write all the information in the paper and then come to the clinic and hand over. But now everything is electronic. Like we started giving them the gadgets, we started giving them the maybe smartphones. Through that, daily they can like report their information, report the data. So samples, like we don't need to the patients or the healthy volunteers, they are showing more interest to participate into the clinical trial because they don't need to come to clinic, they don't need to travel. So there are like lots of benefits like that. And as I mentioned, like targeted medicine are coming, personalized medicines are coming. I have seen one of the student, I think here asked a question about if we are developing the medicine, personalized medicine, then economically how that will benefit. Yes, it's a very valid question. I'm not telling we will develop vaccine for each and every individual. No, it's not like that. But it is going to be based on the gene. So we can easily get the genomic sequencing data. Using that data, we will be developing the drug, and that will definitely help a pool of patients. And not only that, the medical sensor data, we are getting lots of electronic records we are getting. Other than that is the now search engine. Using the search engines, because people will be searching through Google about the diseases. Now if we pull the data, people will be checking about the pandemic, COVID-19, right? Because that's what is happening in our industry now, or in the world now. So like that, through the social and search engine, we know what is the interest of people, that we can understand. Then we can think of a product. Okay, come up with a product that will help people during this time. Okay, lots of safety information we can get early through this data, through online. We can easily analyze the safety signals. And as I mentioned, how we are checking that how can we accelerate the drug development process. There are lots of scientific publications available, academic research paper available, the using all those things, the scientific publication, academic research, we can run the predictive algorithm through this data, and then take an intelligent decision about the like how can we accelerate the process of drug development, drug discovery. And as I mentioned, yes, end of the day, we are trying to reduce our cost and time. Like as Shekhar mentioned, I think all of you will be knowing, once we submit the application, that is new drug application, once we do that, the patent is only for 20 years. And the clinical trial, even oncology trials now started taking 8 to 10 years. There are some of the trials I know, like even I have worked for the trials, they are continuing for 15 years. So if that comes to the market, the sponsor or whoever is spending money, the investors, they will be getting only 5 years to sell that product into the market. After that, a generic version will be coming, right? Everyone need to understand there is a business also behind that. Definitely there is a business. If someone is not investing for this R&D, then how new drug will come into the market? So we need to help them also. So definitely there are lots of activities happening from the sponsors perspective. That means from the pharmaceutical companies perspective, from the authorities perspective, like FDA, EMEA, DCGA, PMDA. Everyone is working towards reducing cost and time. So data analytics is playing a major role because data is available now, data is flowing. So checking all the possibilities to utilize that data and reduce the time and cost.
Thank you, sir, for your input. I would also request Mr. Abhishek to share your thoughts on this question, which I have put forward. So if you can speak a few lines on that.
Yes, I will not take a lot of time because I think Ranjit has covered it quite well. But maybe I will just kind of highlight a few things. We all would have heard about Pfizer vaccines for COVID-19, and it's been brought into the market in the record time. And of course, there are many factors behind that, how they were able to achieve that within such a short time. But one of the key reason behind that was also that they relied a lot on using technologies and automation. So the question is about whether we are using technologies enough or not. we are certainly using the technology, but whether the pace is kind of significant, can we do more? Of course, all that is debatable. The other thing which I want to mention is that there's also a lot of changes from the regulatory perspective. Uh, Now you might have heard about something called rolling submission. Traditionally, we used to submit our trials, our kind of results. Once everything is done, all the trials are completed, all the data analysis is done, all the documentation is done. Pharma being a very highly regulated industry, you also need to produce a lot of documentation. But now even regulatory are kind of encouraging pharma companies to submit data. You're producing it in parts so that this drug development process can be expedited. And again, how they're able to achieve that, it's also kind of due to usage of technology. The other thing which I want to mention is about there was this kind of apprehension sometime that maybe it's little difficult to use some of these technology, data mining techniques, digital technologies within lab. But now we have a lot of examples wherein you can see that AI, ML, robotics, these are being used extensively within the labs also. And due to that, the high throughput screening time is kind of reducing significantly. So to conclude the question which you have asked, I would say the pharma and biotech industry is coping well. Of course, they can do it better. Whether there is kind of, like I said, whether the pace is comparable if you compare it with other domains like banking, analytics, aviation, maybe not. But again, things are moving in the positive direction. Thank you.
Thank you, sir. I have a question. It's a layman question, but like this data mining or the predictive analysis which we are doing, from an industry perspective, when we say like in Mr. Shekhar's slide, reproducibility is a big challenge, right? So when we are predicting something or we are modeling something, if that doesn't go, I mean, what has been predicted, that doesn't go well, how does an industry deal with it? That is another question I would like to ask. If Mr. Shekhar, if you can put in some.
Yeah, I mean, it's an interesting one, but at the same time, we also should underline the, understand the or recognize the underlying fact with the life sciences world is that wherever there is a rule, there is always an exception. That's the nature of our business, right? So when we say reproducibility is more from like, if I perform the same step a number of times, my data quality should be such that I should see the exact same results. But now when I'm using this data and trying to ask a question, depending upon how logical and whether I have all the elements or not, in the data on which I am posing this question, my reproducibility and even the outcome itself would actually depend quite a lot on that, right? If there are pieces to it missing, or there are pieces that have been called as like, so-called outliers, then we will not be able to see things that will start showing up in a formal clinical, a post-clinical trial or phase 4 kind of scenario, right? Again, it's more theoretical in nature, right? I looked at something, I saw some adverse event, but I said this happened in like what, one patient now, right? I can always say this is an outlier. Maybe I acknowledge it, I put it that it's an outlier, but that's my population size there was limited, right? My data showed me something like this. Yeah, the risk of this occurring is low, but that is in the context of the data that I have. When I put it out there in the real world, there again, I should continuously keep monitoring. Now that I have seen a certain possibility, I should keep monitoring that real world data to take an action in a more proactive manner. And then when I see a certain trend which trends to agree with what I have made as an one-off observation in my initial assessment, then I should be able to take action on it, to say either alter the label, or if you have to like, put additional directions in there, or to say, okay, this can't really be taken with this particular medicine. I mean, we got to do something. We have to actually look into it further to say, why did this happen? So if I did predictive analytics, it may have showed that, okay, this is just a one-off case, this is not happening, but real world, it started happening, right? It's just with the amount of data and whether for me, it is statistically significant. But what is statistically significant and what can be observed and the possibilities actually change quite a bit once you go into the real world, right? And that's where, my friends Abhishek and Ranjit were also stressing every now and then the aspect of using the real world, right? It can't be theoretical alone. There is wealth of information being made available in the public domain, and that has to be taken into account when I do my modeling, when I do my simulations, and then I can come up with more meaningful insights. So that would be my take on it, into as to why the reproducibility may sometimes take hit.
Okay, thank you, sir. Thank you, sir, for the information. So I have another question also. Like in order for surveillance technologies to play a role in the next phase of the pandemic, potential privacy concerns have to be a top priority. So how to deal with it? Mr. Mishra, if you can highlight on this.
Okay, I think that's a very relevant question. And if you have seen the recent news, the ruckus that this Pegasus spyware episode has created. So this is not the only case. We have seen quite many such kind of cases recently, wherein I think recently there was a case wherein the Air India database was hacked. So these privacy concerns are reality. It's not a myth. And pharma industry, biotech industry, like Shekhar also mentioned, that we have gold mines of data. We collect a lot of data in terms of clinical trial or through other means. But being the highly regulated industry, there are also a lot of checks and balances in place. For example, like from regulatory side, if I talk about FDA, there's a kind of regulation 21 CFR part 21 that talks about the privacy of patients and healthy volunteers. Similarly, many governments have also enacted a lot of laws to protect privacy of their citizens. And in fact, the pharma companies also have processes in place to ensure that the privacy is maintained. And in fact, at least the pharma companies wherein I have worked in, they have always clearly stated that follow either the organizational policy or local policy, whichever is the stringent. So there is always a lot of focus on this maintaining privacy, and also there's a kind of well-defined process, like how to collect data, how to store data, how long you should store data, and how you can discard it. It's also very clearly mentioned and also ensured that for purpose you're collecting data, so your data should be used only for that purpose and not for otherwise. So a lot of focus on this area. In fact, all the employees in a pharma organization, they are expected to undergo this training every now and then. So I will just say that it's a concern. It's not a myth. It happens, but there are well-regulated policies, processes in place by different agencies like regulatory, governments, pharma company itself. So the chances of happening it are remote, but I would also not deny that the stray cases doesn't happen. They do happen also. So that's my take. Thank you.
Thank you, sir. I would also ask the same question to Mr. Ranjit. I mean, what is your take on this regarding the privacy and security concerns, and how an industry should deal with it? Mr. Ranjit, am I audible?
Can you?
Yes.
Yeah, you
yes,
can hear me, right?
Yes,
yeah.
Yes.
So as Abhishek mentioned, in our industry, we have actually process and systems in place to make sure this confidentiality, actually, because we do the clinical trials. I think most of you maybe heard about it, or if whoever is having basic knowledge about the clinical trials, we do conduct the trials like a blinded trials, single blinded, double blinded, like that. That means our patient information we don't share with the people who are participating into, like who are analyzing this data. Most of the time, I think it's very basic. I'm telling about the clinical trial. The patients who are participating into the trial, they doesn't know which drug they received, and the clinicians who are participating in the trial, they doesn't know even which subject received which treatment. Yes, there are some challenges, and we do conduct the open label trial. That means everyone knows, but still, even in open label trials also, when we are analyzing the data, we treat ourselves as a blinded to avoid the bias. Because otherwise, if we know, now, for example, I am a person who is analyzing the data, and if I know that this patient received this medicine, I can change the result, right? I can make it the way I want, right? So we are maximum, like in our industry, we are trying our best to follow it. Lots of techniques are there in place to follow the blindness. That means we doesn't know which patient received which treatment, and we are keeping that confidentiality. And as Abhishek mentioned, we signed the confidentiality agreement at the beginning of the trial, at the beginning of the activities, starting before our activities, and make sure that. But I can tell you, when we are talking about this data in analysis side, more than confidentiality, now the challenge that we are having is about the quality of the data, as Shagar mentioned, because when we are going into this real world analysis and all, lots of data is flowing, but we don't know this is genuine or not, this is the real data or not. And that is one of the challenge in the clinical industry, because as I mentioned, it's patient's life, and it's taking time. When we are talking about this 15 years, one drug is taking 15 years to come to the market. That means this data is like flowing through different people. There are lots of functions involving into the clinical trial. See, just think that when, like, how one drug is coming to the market, or what is happening into the clinical trial, the patients are participating, or healthy volunteers are participating. They are going to clinic. They are providing their information. Everything will be in paper. Then we are converting that into the electronic format. Then the data management will be coming into the picture. They are cleaning the data. They are putting it in nice format. Then it is coming to biostatistician and programmers. We are analyzing that data. We are concluding it statistically. Then it will go to the clinicians, doctors. They will clinically conclude it. So it's the same data passing through different people, different functions for a long time. So we have to make sure the data integrity that time, or data quality that time. And that is one of the challenge now, why this data analysis is not maybe popping up or not having that much of growth like other industries. Okay, so that is the challenge. How can we make sure the quality? Because I know some of the funny examples I can tell you. This one of the organization in India, they have given Fitbit to their employees to make sure their health. Okay, they told to their employees, you wear this, so we can track like how much steps you are walking in a day, how much time you are sleeping, because just to assess. And people started misusing it. They started putting it to they fixed it with the fan. So then fan will, whenever we switch on the fan, it will give a huge number. And then this person is telling, see, this much of time I used it. See my BMI. It's great, right? I'm healthy. I'm taking this many steps in a day. So people are misusing. So we don't know. We don't know how much quality or how much integrity we can give to this data. That's the reason I told you the clinical industry is entirely different. It's not like a finance or some other industry, because everywhere data analysis is happening everywhere. Now we are getting personal loans or credit cards. How the banks are taking decision? They are setting up a model based on the questionnaire. Now, when you are applying for a personal loan, they will be asking you 10 questions, right? Do you have a property? Do you have a vehicle? Do you have, what is your salary? What is your family members, who all are there? How much they are earning? So there will be 10 questions like that. And based on the, like for each and every question, they'll be giving a mark, score, and they'll be setting up the score. And whoever is getting, if for example, if total score is 100, whoever is getting more than 50, they'll give the credit card. to those people because they are confident that these people will pay back. So how they are setting up? Now, when I'm talking about one person, it is very easy. But think about a Citibank, ICICI, HDFC, the global banks, they will be having crores of application in a day. So they set up a model. It's all happening through statistics. A linear model they will set up. Whoever is getting more than 50 score, they will get the credit card or they'll get the personal loan. So they are using data analysis only there. But it's easy. Risk is very less. Because what will happen in a worst case, this person won't pay the loan back. Nothing more than that. But in clinical industry, one of our mistake can lead to a death of a patient, can have a serious adverse event. So we need to be very clear about this data integrity, data quality, more than confidentiality. But confidentiality, definitely, we are putting lots of process in place. And as Vishal mentioned, yes, regulations are there to make sure that. And even now we have this like lots of new techniques, we call it as data de-identification to mask the patient information. So we are following all those things to make sure the confidentiality.
Thank you, sir. Actually, as you highlighted, with more the technology as it is advancing, there will always be a scope for misuse that cannot be eliminated. And statistics and programming definitely will be an integral part, I mean, in the future for most of the industries. I'm sure many of them are following right now, as you all have stated it. So since many students, actually, I can see they have put forward some of the questions. So I would want to take up one or two questions from the student side and would like to, I request you all to highlight on it. Like one of the students has asked about like if the personalized drugs, they may be effective for one individual or maybe for a few people, but may not be for the others. So how can it economically benefit the company? So your thoughts on it, Mr. Ranjit, since you were speaking, can you?
Yeah, I think I have mentioned about that in the earlier discussion. So it's not about an individual. It's based on the gene. So we can get the data of genomic sequencing or the patient's like the sensor data, the medical sensor data or the electronics records. And based on that, we can develop the personalized medicine and targeted medicine. So when I'm telling targeted medicine, more than individual, it's like targeting some specific maybe body part or whatever it is. What is the reason behind that indication, okay, or that disease? So we'll be developing the drug that will exactly aim the target or focus towards target, and that will give more benefits, and that will maybe reduce the adverse events, reduce the other side effects and all those things. Okay, they can, like, you can Google it and learn about it. There are lots of drugs are coming like that. So it's not about the individual, but it's a group of people or group of samples whom we can target based on genomic sequencing.
Thank you, sir. There is another question. I would request Mr. Abhishek if you can highlight or put your information, if you can share with the students. So one of the students has asked that what is the skill or knowledge which could be expected from a fresher who would want to pursue a career in data analytics, or maybe in an industry which is in which analytics play a very integral or pivotal role. So that is the question.
So I think the key skill is that they should be kind of passionate about data. Unless that is not there, maybe it's difficult to kind of build other skills. So whether it's clinical domain, whether it's financial domain, the understanding of data is very critical. If you don't have understanding of the data, maybe you can apply a software will give you some kind of outcome, but whether that makes sense or not, you will not be able to figure out. So I think the most important thing is the interest, the passion, then the understanding of the data. And once I think these two things are there, the other things can be learned quickly. Like I talked about this open source software. And let me kind of tell you that we do hire freshers from different colleges. We also look for colleges who have a stream in bioinformatics and so on. And what we focus on is that when they are doing their projects, whether they are able to understand the data analysis which they have done, do they kind of know what the data means, what the outcome means, and so on and so forth. So those are the few things which I feel are the most important. Other things you can always learn.
Yes, thank you, sir. Mr. Shekhar, there is another question. It's a similar kind of question, but however, students, they have asked that with a BTech MTech dual degree course, what is the career opportunity or scope in this field of data analytics, particularly after a dual degree course in biotechnology? What are your thoughts on this? I mean,
okay, it kind of again borderlines with or extends or builds on top of what Abhishek was telling, right? There are skills that can be built, right? If you look at today's programmers, right? Logic is something that is inherently to be there as a passion. Syntax, you look at a book, you Google, you do something, you can actually write your program. But without logic, you cannot put a framework through, right? Now, apply the same thing here. In addition to logic, unless and until we have a good grounding in science, right? Whether it is dual degree, single degree, or a PhD or anything, right? Unless and until we look at the applied aspects of this science in relevance to the question that I'm asking, right? It may not necessarily help us in using that piece of technology. I'll take a very old and very small example, right? Now, let's say if I'm if I'm an end user and I say I need to actually come up with a predictor structure for a particular protein molecule, right? All I have is information that this is similar to, this is homolog to so and so, right? The first thing, like, if I'm looking at homology, I have to really be strong in my basics to say, what does really homology mean? Is it like, I take a software, put some data in and then run it with default parameters? It will give me some answer which will not even make sense, right? Here, the question is, I first have to understand how closely are these guys related, right? When I say, very close to very distantly related, the number of evolutionary events that may have happened in the due course of time would vary, right? That comes from our science knowledge. If I don't apply and I run it with default parameters, it will give me something which will not fit the objective for which I'm going. So whether we talk about BTech followed by an MTech and all, right, we should always strive to basically have a good grasp at our fundamentals, consider and look at what are the possible applications, how I can leverage this information, right? And the rest of the things, there are free source versions available, there are a lot of tutorials available, and you can start building your passion up by, let's say, if you want to get into analytics, right? There is clinicaltrialsdata.gov.in, I think, right? They have ton of clinical trial data, right? Let me actually download that. Go get R or Python or something, install it, and now start looking at certain tutorials that are available. Start going through it. Use my knowledge and what I have learned in terms of programming on the data that I have sourced to build, right? And that will help me understand and better appreciate this, identify what my weaknesses are, right? But before I sort of go active homing on this particular area, it would also be very important to say, from a career point of view, there are two things that we always look at. Sometimes it is always driven by passion. Some cases it is mixed by passion as well as the corresponding economic benefits associated with, right? What is really lucrative, right? Or if I set aside the economics part of it, right, what is more employable in the industry, right? Is it a very niche skill, or is it something that is commonly being sought? And in something that is commonly being sought, if I'm showing myself as the most exceptional guy, if I build my competence, yes, definitely I'll be hireable, right? To do a quick survey, to look at the various jobs that are being posted in this industry, look at what they're asking for. honest self-assessment to say, how many of these things do you think are there with me? What do I have? What are the things that I can inculcate? Can I build on top of it? Right? Then can I meet? Identify what the direction is going to be, and then start getting into these tools, techniques, and other things to build, to get some hands on, to build that passion, to basically better understand, better appreciate, and all. And I think that's the direction we got to go. But we never, ever, set aside the science. We set aside the science, it's just not going to take us anywhere. A tool can always give us a result. When we say actionable insights, it gives me some insight, but what action to take on it is purely dependent on the science and the underlying facts that I know about how to assess that. Tool is not going to tell me end of the day to say that you go ahead, switch off this fan. It will show me there is a higher energy consumption happening. I look at the devices and say, okay, this is the guy that is consuming more, because this is like a 40-watt unit compared to a 9-watt LED bulb. So I will go ahead, switch it off. That decision I have to make based on the knowledge that I have. Right? And that is application of what I am learning in my BTech, MTech dual degree courses, or even BTech followed by any diploma courses, etc. That I think is going to be the real key, the real underlying thing, right? Science cannot be taught. IT technology can still be taught to a person, but science, the underlying, the basic sciences cannot be taught, right? I can always tell an IT professional how to code, or I can learn how to write a program. But a programmer, I cannot really teach the central dogma and then say, this is exactly how it works. This is the technology of DNA sequencing. These things are difficult. They are grounded. They are trained in that life. So that's easier understood for them. For us, we are trained in science. And we got to basically use that. We have to leverage it. Right. So that would be my take on it. That is something that not just preaching, I've been practicing it as well to like, that's how I'm making my career for the last 16 years. I use my science understanding. I use that language that I speak in various laboratories across the world. I see and understand how the process works. I understand what is the regulations. And then I tell my IT folks that this is exactly how our system should behave. These checks are valid. These checks are invalid, right? All of these things, right? So it is really the science, I would say, without any hesitation, for the last 16 years, my basic sciences of genetics and bioinformatics has been driving my career. That's it. Rest everything is experiential, and everybody in this world can gather this experience once you get into that line, right? With that, I would probably stop and like to hear from others.
Yeah, just to add my sense here, see, generally what we see is whoever is learning mathematics, they doesn't like science or life science, I can tell you. Because now I'm a mathematician by education. I have masters in statistics. So I was not enjoying my botany or zoology classes that time. Okay. Same way, whoever went to the life science side, the biotechnology or microbiology, whatever it is, they doesn't like mathematics. They always tell maths is very tough. Okay, this is what we can always see. Now, when it comes to a data analytics, and especially into the clinical industry or pharmaceutical world and all, it's a combination of both. When I started my career with Novartis, because I have completed masters in statistics, Novartis came to my campus, they have recruited me as a statistician, and I entered into this industry. I was wondering what statistician will do in a clinical industry. And they started telling me first day onwards, this protocol, statistical analysis, placebo, crossover, parallel. When I heard placebo first time, I thought this is something weird. So it's all we can learn. It's as Shekhar mentioned, or as others mentioned, it's we can learn that. Don't think that it is impossible. No, you can. And try to relate with whatever you are seeing. Now, every day, definitely every day you are brushing, you are using toothpaste, right? How this toothpaste came to the market? There is some data analyzed behind that. You are using soap. Now, when I'm talking about the paste, soap and all, these all are the health product you are using, right? As a human, we are applying it into our body. After using toothpaste, tomorrow onwards, if you are like breaking your teeth, you are losing your teeth, or it's creating smells, it's becoming yellowish. You are not going to be happy with that toothpaste, right? Then why we need that type of toothpaste into the market? So how that came into the market? We have already analyzed that product. We have given to some people, maybe 100 people we called. We have given 50 that paste, 50 to some other paste or some other product. Then compared, then it came to the market. So it is happening everywhere. Whatever products you are using now, daily, it has gone through this period. So you can easily relate this data analytics, and don't restrict data analytics to clinical industry, as I mentioned. No, it's happening everywhere. Anything coming to the market, it's through data. So that's what you are going to do. And don't think I'm a science student, I can't learn mathematics, or I'm a mathematician, I can't learn science. No, you can. When it comes to an experience or the application side, definitely you will enjoy it. Seriously, I'm telling you, will enjoy it. Early days, even I struggled, but I'm really now as a statistician, I can even go to banking. I can even go to insurance. Anywhere I can go. Statistician is having lots of value. But why I'm not going? I'm in this industry for 16 years because I really enjoy that. And we are not getting, seriously, I'm telling, in a year we hire around 30 to 35, means for my organization, I'm telling, in a year we are hiring around 30 to 35 freshers for a data analysis. We hire life science students because it's easy to teach them science. It's easy to teach them pharma industry. And we hire statistician also. So we hire everyone. We are very confident. If you have that smartness, if your communication is good, if you are ready to learn science, if you are ready, if you are ready to learn, if you love numbers, little bit of science knowledge, it's absolutely fine. You can build up the career. I'm telling you, just go. I'm sure in LinkedIn, Instagram, Facebook, and all. I don't know how many of you have profile in LinkedIn. If you don't have, please go and create it, because you all are looking for a career in future. In LinkedIn, you just search data analytics job. I can tell you, will find 1 lakh of opportunity, lakhs of opportunity for a data analyst. So that is the way our industry is heading.
Thank you, Mr. Ranjit. Actually, whatever you told is absolutely true. Probably our research we have to integrate with other disciplines, and probably that is what industries want. And as you told, Mr. Shekhar, I agree that there is no alternative to fundamentals and basics. So students here also, they think that many of them have in their mind that, like you said, that if I'm a biotechnologist, I might not be good in mathematics, but learning, there is no limit to it, and everybody can learn and do and integrate. So I thank all the 3 speakers. Actually, it was a very, very informative, and we had a very nice, interesting session. And thank you all for taking out time and addressing us and sharing your views and informations on data analytics. So it was very valuable, and we had a good discussion. I would, we now come to the end of the second session for the first day of the Industrial Conclave. And I thank my fellow faculty colleagues and all the students who are present in the session. So before ending the session, I would want to make an announcement that all the students and faculties, please do join for tomorrow's session as per the time and Zoom link, which has been shared by Dr. Gopal in your respective email IDs. So with that, I would like to end it. Thank you all and have a good day.
Thank you.
Thank you, Gopal. Thank you, Amrita.
Yeah, and also if there are many kind of unanswered questions, maybe you can mail it to us and we will see what we can answer.
Yes, sir. We'll definitely do that, because many questions are actually remaining which were unanswered due to lack of time. So we will be definitely sending you those questions from the students. Right. Okay. Thank you, sir.
Yeah, that will be good, because I can see good relevant questions about crossover designs and all.
Yes.
Definitely we will answer it. And thank you so much for this opportunity and helping us to connect with the next generation.
Thank you, sir. Our pleasure.
Thank you all.
Bye.
Have a nice day. Bye-bye.